Good afternoon, everyone. I would like to welcome you all today to the section 2, stem cell therapy in clinical application, current status, and new perspectives. My name is Kureysh. I'm the head of the Department of Normal Physiology at Astana Medical University. I'm sure you all know Tatyana Strekalova, University of Maastricht, Neuroplast BV, Netherlands. In today's meeting, we'll cover 4 research reports dedicated to stem cell therapy. So let's begin. It's my pleasure to present our first speaker, Professor Vyacheslav Ogay, Deputy Director for Research, National Center for Biotechnology, Astana, Kazakhstan, and his research the therapeutic potential of mesenchymal stem cell in the treatment of atherosclerosis. Please.
Dear colleagues, good day. So my name is Professor Vyacheslav Ogay. I'm Managing Director for Science at National Center for Biotechnology. Last year, I was just head of the laboratory for stem cells, and today I'm presenting my research results on application of mesenchymal stem cells for treatment of atherosclerosis. That? So in this presentation, we consider what is atherosclerosis, current therapy of atherosclerosis. We'll talk about mesenchymal stem cells and therapeutic as MSC in atherosclerosis, strategies to improve therapeutic effects of MSC, and I will present our preclinical data on the efficacy of MSC therapy for atherosclerosis treatment. So what is atherosclerosis? Just little memory about this disease. So this atherosclerosis is a chronic inflammatory disease of the arterial intima, just characterized by formation of atheromous plaques, leading to occlusion of arteries. So you can see here this atherosclerosis plaques inside the artery, including the thrombosis. So atherosclerosis is core of cardiovascular disease that cause several important complications like ischemic stroke, vision loss, heart attack, heart failure, kidney disease failure, and sexual dysfunction. What is risk factor of atherosclerosis? So about risk factor, diabetes, smoking, hypertension, obesity, age, and genetic abnormalities. And if you see these slides, we can see the epidemiological data. So disease is more prevalent in men than women. About prevention and treatment, so the patient has to be must stop smoking and drinking, and he has to be make exercise and keep the diet. Okay, I just pass this slide. About current therapy of atherosclerosis. So the current therapy of this disease, first one is surgical treatment, like elimination of atherosclerosis plaque by open vessel surgery, balloon angioplasty, and coronary artery bypass grafting, and artery prosthetics. Second therapy is drug therapy. So nowadays, the doctors use statins for reduction of cholesterol levels. So unfortunately, approximately 2/3 of patients under the treatment of the statin continue to suffer from the expected cardiovascular disease events. And many patients cannot tolerate statin or follow along the term statins treatment to reach optimal LVCC level. So nowadays, the alternative therapeutic approach, this mesenchymal stem cells. So mesenchymal stem cells is, this this is adult stem cells that can be isolated from different organs and tissues like umbilical cord, bone marrow, fat, placenta, and so on. Mesenchymal stem cells possess immunomodulatory properties. So mesenchymal stem cells affect all immune cells like T cells, B cells, NK cells, macrophages, monocytes, dendritic cells, and neutrophils. They produce cytokines and growth factors like PG2, IL-6, TGF-beta1, IDO, and so on. So according to this literature data that was published in the Cell journal, there are two types of mesenchymal stem cells. So it depends on inflammatory environment. So if mesenchymal stem cells sorry. So if mesenchymal stem cells will be treated by high-level inflammatory cytokines like interferon gamma and TNF-alpha, so they polarized to MSC2 phenotype. And MSC2 phenotype, they are anti-inflammatory cells, and they produce high-level IDO, NO, and PG2, and also TGF-beta1. And they also activate T regulatory cells, and they suppress the inflammatory process in the body. So if mesenchymal cells will be treated by a low level interferon gamma and TNF-alpha, they transform to MSC1 phenotype, and they produce low levels of IDO and PG2, and they produce some chemokines like CXCL9 and 10. And in these immunomodulatory molecules, they activate the T cells like T helper 1. Th1. Here we can see another model for interaction between MSC and monocytes. So MSC balance polarization on monocyte to M1 and M2 macrophages. So M1, this is they produce inflammatory cytokines, and M2 they produce inhibitory cytokines for suppression inflammatory process. So in this slide, we can see that MSC can affect on different populations of immune cells like Th2, Th7. For example, MSC can inhibit functional activity Th17 and activate another population, Th2. This is T suppressors lymphocytes. And for other sides, also activate T regulatory cells and block activity Th1. Regarding macrophages, so they block activity of macrophages to form cells during atherosclerosis. From other side, the MSC can affect on macrophages and transform them to macrophage M2, this anti-inflammatory type of macrophages. And here we can see the several therapeutic effects of MSC during atherosclerosis. This picture was collected from some literature data. And MSC can affect on endothelial dysfunction, so they can improve endothelial dysfunction, inhibit inflammation, reduce dyslipidemia, reduce foam cell formation and plaque size, enhance plaque stability, inhibit vascular calcifications. So from this slide, we can see that application MSC cause beneficial therapeutic effects during atherosclerosis. So here we can see the table of preclinical studies using MSC for the treatment of atherosclerosis. This data was collected from several original papers that was published since 2015. And we can see this animal model, this OPOE, this is transgenic mice, LDR, and one model for rabbit model with atherosclerotic plaques. And applications for cell therapy, they use, for example, mouse skin and human MSC, mouse from bone marrow, or human bone marrow. And from this side, in this cell, we can see therapeutic outcome after application of this mesenchymal stem cells. So we can see that MSC induce very good beneficial therapeutic effects during treatment of these mice for rabies, for example. It's just example from this paper that was published in Molecular Cell Biochemistry, 2015. They observed that mesenchymal cells elevate atherosclerosis by elevating the number of function T regulatory cells and inhibit macrophage foam cell formations. This another paper was published in scientific reports and also about 8 years ago. And we can see that intravenous application of mesenchymal stem cells just reduce murine atherosclerosis development. They reduce level of interferon gamma and TNF alpha after 8 weeks. But from other side, they increase level IL-10 and IL-6. Histological analysis revealed that MSC administration also reduce size of atherosclerotic plaques inside the artery of these mice. About strategies, how to improve the therapeutic effects of MSC, because before I showed just several examples that intravenous administration MSC causes therapeutical beneficial effects during treatment of atherosclerosis. So how to improve the therapeutic effects of MSC? There are several strategies. First one, MSC can be treated in hypoxia conditions or treated by inflammatory cytokines to induce anti-inflammatory mesenchymal stem cell type. Another strategies, we can change the cultivation conditions, and we can cultivate MSC in like 3D cell culture. They form the 3D spheroids, and so in these physiological conditions, they increase the inflammatory, immunomodulatory, and inflammatory properties. So another strategies using genetic engineering, we can modify MSC. For example, we can insert gene, for example, IL-10, and they produce this immunoinhibitory cytokines to reduce inflammation. So in this slide, we can see our preclinical data of the efficacy of MSC therapy for atherosclerosis treatment. We have a research grant, and I will show our results. So the purpose of our study was to evaluate therapeutic efficacy of TNF preconditioned MSC using animal model atherosclerosis. And first task was to successfully isolate and characterize mouse compact bone MSC. Second task was induce atherosclerosis in knockout ApoE mice with Western type diet. And third task, preconditioned MSC with pro-inflammatory cytokine like TNF alpha. And last task, we evaluate efficacy of intravenous administration of TNF MSC on development of atherosclerosis in ApoE mice. And here we can see outline of our preclinical study on evaluation of the efficacy of TNF MSC using animal model atherosclerosis. So we induce atherosclerosis using Western type diet. So we purchase this how to say food. Yes. Yeah, food for this mice from Germany, the SNP company. This will contain 0.15 cholesterol or 0.21% butter oil. It's very fat food. And we feed our animals during 8 weeks. So after that, we inject MSC. This intravenous injection 2 million cells per mice, and inject 2 times, and interval was 7 days. Here we can see the groups. Control group, just PBS injection, MSC group, and TNF preconditioned MSC. And we use ELISA, flow cytometry, microscopy, and histological analysis to study how TNF MSC affect on the development of atherosclerosis. According to our task, we successfully isolate and characterize mouse compact bone MSC. So we can see here the primary culture of MSC under phase contrast microscope. This is just bone chip, and here isolated MSC. And this primary culture of MSC. This is CFU assay. We evaluate the clonogenic activity of MSC, and we differentiate them to adipocytes, osteoblasts, and chondrocytes. Also, we studied phenotype of our MSC culture. And we can see that they significantly express CD29, CD42, CD90, and CD105. And they don't express CD31 and hematopoietic cell marker CD45. So for this study, we obtained 3 primary cell cultures. And also we tried to get 3D cell culture of MSC. So here we can see 3D spheroids in our culture of culture plates. And this is CLSM image after staining by CD19 and rubidium iodide for staining nuclei. This orthogonal sectioning, so we can see that spheroid has no any cavities. So by using CLSM, we evaluated the quality of our 3D spheroids of MSC. So in this slide, we evaluated the production of immunomodulatory molecules like PG2 and IL-6 and compared between 2D and 3D spheroids after treatment by TNF-alpha and interferon gamma individually and in combination. So our conclusion was that TNF-alpha was more effective and suitable for preconditioning of MSC. And we further use TNF-alpha for our experience. Why we don't use interferon gamma? Because according to literature data, after treatment interferon gamma, MSC, they highly express MHC1 and MHC2 molecules. So the immunogenicity was increased. So for this reason, we selected just TNF-alpha MSC. So our second task was to induce atherosclerosis in knockout ApoE mice with Western type diet. So after 8 weeks, we sacrificed several mice and examined the artery, the coronary artery. After staining oil red, this orange, the spots, here we can see the plaques in the artery. So from this experiment, we proved that we induce atherosclerosis in these mice. So after that, we started application of our MSC according to our preclinical outline, our preclinical design. So after 2 injections, 7 days intervals, we evaluated cytokine profiles in peripheral blood atherosclerotic mice after treatment by MSC and TNF preconditioned MSC. So we evaluated the levels of TNF-alpha, interferon gamma, IL-6, and IL-10. And we observed that after treatment by TNF-MSC, the level of TNF-alpha in peripheral blood was significantly decreased if you compare between control group and just MSC group. And also observed when we analyzed interferon gamma in peripheral blood after 8 weeks. IL-6. So ELISA revealed that after treatment by MSC and TNF-MSC, the level of IL-6 was increased compared to control. So IL-10 also was increased, but significant difference between MSC and TNF-MSC we didn't observe after 8 weeks. So another experiment was to evaluate how TNF-alpha MSC affect on cholesterol, LDL, and HDL in blood plasma in our experimental mice. And for this experiment, we use commercial kit from ECAM to measure total cholesterol, HDL serum, and LDL serum. When we measured total cholesterol, we observed that total cholesterol was decreased in two groups, MSC and TNF-MSC. Unfortunately, we didn't observe significant differences between these groups, but they differ from control, significantly differ from control. Regarding HDL, high density lipids, we observed significant increase in TNF-alpha preconditioned MSC that was significantly higher than after treatment by just MSC. According LDL serum, so LDL serum was decreased after 8 weeks significantly, but between two groups, MSC and MSC treated by TNF-alpha, so we didn't observe significant differences. So when we finished our experiments, in the experiment, we performed necroscopy and we isolated spleen. So here we can see the control mice spleen and PBS treated after PBS treatment, MSC treatment, and TNF-alpha treatment. So PBS was like sham control. So we can see atherosclerosis induced increase of size of spleen because during atherosclerosis we observe inflammation in this organ. So after treatment by MSC or TNF-MSC, the size of spleen was normalized compared with control and compared with PBS treatment. So we can see in this graph that's big difference between groups. And also we made histological analysis stained by chemotoxin resin and oil red to differentiate atherosclerotic plaques inside the artery. So we can see in PBS treated group, the plaque was very big, very large size. So after treatment by MSC, the size was significantly decreased. And after TNF-alpha, we also observed significant decrease of this atherosclerotic plaques. So the size of this atherosclerotic plaques we can see in this graph also. So if you also we check the appearance of atherosclerotic plaques after MSC therapy. So we can see in TBS group, the appearance was not very bad, and the hairs, the back, and after treatment MSC and after TNF-MSC, the appearance of atherosclerotic plaques was significantly improved. Is our conclusion. So recent animal studies have shown that MSC exert beneficial effects on reducing atherosclerosis development by the immunomodulatory and anti-inflammatory properties. Preconditioning approach significantly enhance immunomodulatory and anti-inflammatory properties of MSC. And we have shown that preconditioned MSC exert more powerful immunosuppressive and anti-atherogenic effects in the development of atherosclerosis compared to the untreated MSC. And we believe that the use of preconditioned MSC can be recommended for developing a strategy for the prevention and treatment of atherosclerosis. Thank you.
Are there any questions?
I have a question. So if your culture MSc is in spheroid form, will they tend to spontaneously differentiate on like how susceptible they are to spontaneous differentiation?
For in 3D culture, we just cultured just several days, and we didn't observe any differentiation of MSc during this cell culture conditions. Just 3 days. So after that, we have to apply our MSc for cell therapy or for another eggs.
Do you make a spheroid in a bioreactor or special vessel?
We use special vessels with low binding surfaces.
Okay. Thank you.
Thank you. That's such a slide.
Why we didn't use 3D steroids? Because when we inject inside, so we use like intravenous administrations. So we can encounter with some complications. Thrombosis. Да, thrombosis. Yeah.
My question, did you observe the fate of your mesenchymal stem cells after injection? How do you know that they go exactly the place you know before? How long they stay in the body of the mice after injection?
So unfortunately, we didn't start this fate of after injection MSc. We just observed the level of inflammatory or inhibitory cytokines, and that's all.
You can do it basically. You can use our method and try to do it. We can modify it. We use the GP protein, inject, you can see if you want to do it. We can modify surface. Just interesting. They're going to block, so they just produce something with a block. That's it. That's it. Mechanism of action.
So in this experiment, in this study, we observed that TNF5 pre-conditioned MSc, they're more effective than untreated MSc. So it means our hypothesis that we increase immunomodulatory properties of this MSc.
My last case is MSCs don't go to the site of the injury, they just produce something, maybe more.
Yes,
it's a more safe approach, we just produce the condition video.
Or exosomes sometimes.
And inject exosomes video with cytokines instead of cells. This approach kind of safer and cheaper.
Yeah, I agree with you.
Yeah, so I think it's very important to see if they go there and do something there by themselves, or just they produce something and this production does something.
Yes.
Ask a very quick question. So in the beginning of your talk, you mentioned that you can also use hypoxia, if I understood this correctly, to stimulate.
That's one of the strategies. Yeah.
Right. But can you try to actually use it, and why did it choose rather TNF?
For example, for hypoxia, we didn't make this experiment. And why we decided to use inflammatory cytokines? We just based on previous nutrition data. For example, if we treat we treat our stem cells by high level inflammatory cytokines like TNF5 or interferon gamma, so we can get anti-inflammatory phenotype MMC. So based on this data, so we select this strategy. Yeah. Yeah.
Already existing.
Maybe a question. Hypoxia, if you see that stem cells are kept under low oxygen levels to stay neutral, to not influence. What do you think if you treat them with hypoxia, that you get another phenotype? Because I think if you stimulate the MSCs with hypoxia, that you get more of neurovascular activity than an anti-inflammatory. And then, of course, that can both help in atherosclerosis. It is always the oxygen level by occlusion, and on the other hand, the inflammation. What do you think? Can oxygen of low oxygen really help, or is it more keep the MSCs not in an expanding mode?
I think if you use hypoxia for preconditioning or MSC, so probably we also target some improvements, for example, in the angiogenesis. In our research was just examine how TNF or interferon can detect or improve the immunomodulatory effects of this MSC. So we can check next experiments how the hypoxia or just combination hypoxia and intermediary cytokines like TNF alpha work together. Yeah. So that,
Thank you. Okay, we can continue on the next item of our agenda.
And it's my great pleasure to introduce Dr. Johannes de Winter, who is the chair of neuroplastic company, and this company specializes on the use of stem cell therapy, spinal cord injury, for example, and does a lot of basic research that supports the leadership in these studies. So Johannes will present us the talk, stem cell therapy in models of traumatic spinal cord injury and neurodegeneration, advantages of standard therapies. Strategies.
Thank you all. Thank you for the invitation. Can you hear me on the back? Yeah. I have a very loud voice, so I do not use the microphone. Okay. It's autologous, so that means that the stem cells I'm going to present are from the patient itself. It's not modulated, not IPSs, only the patient's bone marrow. What's available, I will show. I do a short introduction, give you a backline of the company, what I do. Then I want to address something when you go to the market and you want to do something, really go for patients. And you have to follow in a certain strategy, I will tell you, the pathophysiology, then the preclinical program, and in the end, and that's, I think, I'm happy to do that, we have some patient background. So I want to show you the idea, manufacture, preclinical modeling, and last, going in patients. Now, this is my life. I started as an emergency health physician, meaning every accident I was dispatched. So I met my first spinal cord injury patient, which I treated very well. I was very happy that he got him alive in the hospital. And if you discover 6 months later that he threw himself for the train because he could not live with spinal cord injury. I was young, so it has an enormous impact. It took a while to do something on it, but at last I found something with less care. That was right, and in between I did my PhD. With the help of a lot of people. This is where we're located in the south of the Netherlands. And you see, it's in the center of Europe. Germany, Belgium, and here is France, and there is Norway. So I think it's a good location. I like it very much. It's a small industrial site. We are situated in a very old building that's built in the Second World War. to produce chemicals for the Germans, and now I do stem cell. It is now used in a decent way, and we have a new facility in Maastricht. This is what I wanted to show you first, because I see that some physicians are here, I see some engineers are here, and in the long run, everyone wants to demonstrate something that you go really in patients. But it starts in the designing. So if you want to go in the patient, and I think that the colleague speaking before is going to do something on atherosclerosis, and he wants to go, of course, in the clinic, because that's what we need, those knowledge. Then you start at the beginning with your design. Be careful that you use materials already in a certain level. It is mostly possible to use research grade. Don't do that if you really want to go straight to a human trial, because later on you have to change that. And this is the whole part with a lot of decisions, waiting, approvals, frustrations, and now we are there. This is what I wanted to tell you, and I think that it is some similarity with the speaker before, because I'm also addressing inflammation. You know that a spinal cord injury happens, there is a disruption of the blood-brain barrier, there is a lot of inflammatory cells, and the lesion itself, don't cite me everywhere, but it is around 10–15% of the original lesion is caused by the damage, and the rest is caused by secondary inflammation. Because the whole inflammation is worsening the situation. And that's what we would like to attack with the cells. And that is in the acute and the subacute phase, where you try to avoid the astrocytes clotting to a scar, protecting the tissue, to activate the microglia to type 2, to the more regenerative potentials, to myelate the damaged neurons, and hopefully more. But one of the problems with stem cells is that they are not really focused. If you work with stem cells, and in general you let them loose in an acute environment, they behave to stimulate or inhibit the inflammation. But on the other hand, if you let them loose in a more chronic situation, then you different thing, the same cells. That was the question, the discussion we had just before about oxygenation and inflammation. But stem cells can do more. They are multitaskers. Because normally women are multitaskers, but in this case, stem cells too. And that's why I see them as the chameleon under the cells. They adapt very fast to their environment. And don't underestimate that, because you lose your potential to heal. What's in our products? It's called neural cells, not because there are neural stem cells in it, no, because we use it for neural indications. And the most important parts are hematopoietic stem cells, mesenchymal stem cells, and endothelial ones. And what you see here in this tree cocktail, you see that the hematopoietic cells influence inflammation. Mesenchymal cells regenerate, influence immune depression, can act in a good way, but together they can enforce the working of anti-inflammation, and the endothelial ones are mostly restoring the oxygenation of the spinal cord injury. Because in the spinal cord injury, there's also a disruption of blood vessels. There is ischemia. It is a multifaceted disease. Mode of action, there you see them again, interleukin 6. That's also mentioned by the speaker before me, TNF alpha. What we have demonstrated in animal studies with the help of Tatyana and Alexia, with the help of you. We look carefully in the cells, and we found that the cells lower into the anti-inflammatory interleukins, and that they stimulate macrophages and microglia to get more in the good guys. Normally, when they are activated, they are very aggressive, hurting the tissue. But if you can polarize them, they get more friendly, more regenerative, more healing. And what you see there, that is a provocation test where we put the cells in a control situation, activate them, and see that they lower the interleukin produced by monocytes. Preclinically, we did safety first and tumorigenicity, because I don't know how it's here, but in Europe, your own cells are the most poisoning cells you can use. It is really terrible to use stem cells. Everyone is afraid of getting tumors. And regulatory says, oh, what are you doing? This is wrong. It's a very dangerous material. Nothing happens. The cells did not get tumors of the animals. We did tumorigenicity, and it was shown that it protects against tumors. We did a balloon compressed model with T-cell deficient rats, that we did with the University of Maastricht. We did a head-to-head comparison with the product and methylprednisone, because the only therapy for spinal cord injury, the acute one, is a high dosage of steroids. So we thought, let's compare who's better. We did dose relations, we did biodistributions, and that showed all in the right direction. This is an overview of the preclinical data. First, the animals get less complications and did less number died compared to the vehicle-treated animals. We had small effects in hind limb functioning that was shown in animals. Small effect because we use T-cell deficient rats. And if you immune modulate and you use a model which has an impaired reactivity of T cells, then your results will be less, because we were at that time afraid that human stem cells in a rat will have a wrong combination, will get rejection, and then the cells are gone. It was not the case. Then we did a comparison with prednisolone. You see the difference. TNF alpha was increased in the vehicle, not in the treated neurocell. Interleukin beta we did. And there, the 6 is that we compared the polarized macrophages microglia in the tissue of the rats compared to the neurocell treated and the vehicle treated animals. So there you see a higher degree of polarization. Then the phase 1. Before you go to patients, you first want to demonstrate that the sample and that it is safe, even if that are your own cells. That was a different one, because what is a healthy volunteer? Because that's a definition. And you can imagine if you're sitting here and I ask you, may I take some bone marrow out of your hip, and you check that intuitively in your bag, that everyone says, yes, do that. No, that's not the case. So we selected chronic patients who had a lesion of nearly 3.5 years. We did motor functioning, which that means that you have an assessment where you can demonstrate how powerful are your muscles, can you do anything if I provoke you. And then we followed them for 3 months initially, because we wanted to pilot phase 2. And in the end, we followed them for 2 years without any side effects. And we tested the chain because this was a fresh product, meaning I take it out of your hip, I fly it to my factory, I produce it, I test it, and I fly it back, and that all within 48 hours. And Toledo is like, I think, from Astana to Moscow, and then by plane. So you first collect it, and that's quite an operation, but it was possible, even in Spain. This is the testing what I wanted to show you. You see here, this is the muscle testing, and that means that if you can, for instance, if you can do this, you have 5 points on your finger. But if you cannot do that, and I have to help you, then it's 2 points. And so all the muscles of the upper part and the lower part are filled in, and here's the feeling. Do you feel anything if I touch you? And if I take a needle, hurt. And that are the testing. These are the results of the phase 1. We see that we have some changes in sensory. We have some changes, but this is, I think, more coincidence in motor functioning. And we have some changes in degree of the spinal cord, from a complete lesion to a non-complete lesion. That means that there is more autonomic functioning. In principle, it was safe. No one died, everyone was happy. I was happy because no one died. So the phase 1 was good, and it gives some information. Imagine that you are sitting 3 years in your wheelchair, doing nothing, because therapy is over. Your muscles are completely gone. And I inject you something which can stimulate the reconnection with your muscles. But if you have no muscles, how can I see that? Because the tests I show you, let's see that you can do something with your fingers or your legs. That takes a long process of rehabilitation, and that was not included. We only injected, nothing more. So if I want to do it over, I would combine it with a rehabilitation program. But things happen. The green ones were the, if you can see that, that's a little green. This one is the only patient got worse. The rest, the green one had a autonomic functioning. And then the circles I show you is where you have more feeling. You can imagine that you don't feel anything. And after the treatment, you feel a normal feeling in your skin. Is it a big improvement? For patients, 3 years waiting, I think it is promising, but we are not there. Then we did a randomization and placebo control, because that is what you need to do when you want to demonstrate efficacy. We did a whole range of testing, and we did an early administration, and we did a late administration, because in the ethical committee was demanded that the placebo patients got the treatment afterwards. Now you deep into your mind, it is designed for acute. And we try to organize it in a way where rehabilitation is there. So we started in a time frame that the diagnosis of spinal cord injury is secure. Because sometimes you see spontaneous progression in a good way, but that was anyway done. Because swelling is lowering and your outcome is better. These are the results for the interim result. 3 months. We are just measuring, so it is not complete. But we see that sensory is good. We see motor function increase.
And this is the only data I can show you now because it is unblinded last week. So this I have the 3 months data, the 6 months data are under investigation. But what you see is that it has some effects, small. Now I'm looking at the 6 months. Is it a permanent effect, or is it going back again when the cells are out of working? But this is promising. And this, ladies and gentlemen, is what I wanted to present to you, to give you an overview that stem cells can really do something, but we are still not there. The next plan, what we have in common, is that we go to the early stage, that we do 24 hours after the incident, collect the bone marrow, and then give it back 48 to 72 hours after the incident. So that we are in the real middle of the inflammatory process, getting down. Because you can imagine, if I'm standing at the Chinese border and I'm one soldier and I want to keep the Chinese in China, then if I wait too long and all the soldiers of China are in move to the border, then that one cannot stop. Then you get overwhelmed with people, and that is not possible to handle. That's a chain reaction. But if you are at the early stage, that the chain is activated, maybe you can do with 1 or 2 cells, in general, stop the cascades. And then you have to go very early. That's the next stage what we are going to do. Questions? Not too much?
Basically, question. So I understand you use bone marrow mononuclear fashion. You don't separate like hematopoietic stem cells or medical cells. It just make it without any modifications. And my second question: did you consider to use adipose-derived stem cells? Because it's easy to get, it's less invasive operation. People usually get easy that way. Why asking? Because what I did myself, just to share that story, I put platform here and but it purified and injected to my knee because I had arthritis, not arthritis, arthritis. And now it seems to me it's improved. 2 years now, and I have no occasions of course, it's not like a scientific job. I decided to do it by myself because I have opportunities to do it for treatment for people. And so I'm thinking that adipose-derived stem cells could be a good alternative to bone marrow cells.
That's a very good question. Do you need really bone marrow cells? Mesenchymal cells are everywhere, and this is true. Adipose is a rich source of mesenchymal cells. But I want first answer the question. Why did we take mononuclear cells? That's quite simple. And that is, I think, crucial for everyone here with some cell experience. A stem cell is what is a stem cell? We are talking about mesenchymal cells if they adhere to plastic, if they have line differentiation in 3 lineages, and if they have certain markers. To be honest, that's not a mesenchymal cell. That is what we thought were mesenchymal cells when we did the definition. But that's not a mesenchymal cell. And if you look to a mesenchymal cell, that cell doesn't do nothing. That's lying and waiting. It's a progenitor. That is a precursor cell waiting until it's activated. And what we did in the whole procedure, and that is not the easiest one, is keep those cells untouched. Because normally you can label those cells, extract them, or you stimulate those cells from the fraction to adhere to plastic. But then you have not a naive stem cell anymore. You have an activated stem cell going to expandation. And that's not the cell I need. I need a virgin. I need a cell has a clear mind, makes the decision when it enters the body. That's crucial. And you see the failures. If you look at literature, you see a lot of failures with all kinds of trials because we wanted to give numbers. We are focused on numbers. Give 2 million, no, 10 million, no, 100 million. But then you don't understand the concept of cells. And I think the prior speaker talked about cell-cell interaction, talks about extracellular vesicles, talk about cytokine. Does the stem cell produce the cytokines? No, it stimulates the environment. As it can stop production, it can also activate it. It's a decision-making cell. So keep that cell as neutral as it is, because if you take that cell out of the low oxygen that it gets in the bone marrow, then it stays calm, lazy, sleepy. But as soon as it gets oxygen, it's going to expand, differentiate. And that's not what I want. Adipose, yes, it could be a good tissue, but it is also proven that you can form neurons from adipose tissue. But the procedure we did at the moment was that we wanted to immune modulate, and we think that is only thing that bone marrow cells are designed to stimulate immunity, but that they are more powerful in the long run to influence immunity. But this I have no proof for that. So that will answer all your questions. Other question?
So what dosage of MSC did you use? Dosage.
Dosage.
Yeah.
As much as they can get out of the body. Because you are limited. We have a release. We stated the release of the cells that at least 4 million hematopoietic stem cells are needed and 4 million mesenchymal lineage cells. That is what we state. In general, we are around 7 million of each. But it is what you get, because I'm not expanding the cells. I take it out of your hip or your hip. I'm more pointing to you, but there are more hips there. Because I take it out of the hip and try to clean it up, take out what I don't need, because it's for intra-articular application. I don't need erythrocytes, since I get them out. I don't need monocytes, TB cells, and natural killer cells, so I take them out. And then the remains are injected. Yeah.
Right. Okay, I think. Thank you very much for the long presentation. I think we can go on to the next speaker.
I would like to present our next speakers, Professor Maladek Askarov and Dr. Anastasia Ganina, and National Scientific Medical Center, Astana, Kazakhstan, and their research stem cell therapy through systemic lupus erythematosus. Please.
The one who's presenting this presentation? Yes. Нет, второй плохо слышно? Я думаю, так и будет. Да, ну смотрите. Good afternoon, dear colleagues. We present presentation about stem cell therapy for cyclic lupus erythematosus at the National Scientific Medical Center. So what is the lupus erythematosus? It's systemic lupus erythematosus is an autoimmune disease characterized by antibodies to nuclear and cytoplasmic antigens and replacing and remitting spores. Our study design consists three criteria. First is the selection criteria, is reliable diagnosis. Second, the activity of the autoimmune process. Two, or pedigree, lack of effect of previous basic immunosuppressive therapy, patient ages from 18 to 55 with resistance to immunosuppressive therapy. Group characteristic is the patients were divided into two groups. The main group is stem cell therapy, 53 patients in combination with high dose immunosuppressive therapy. And the control group, 50 patients. Scheme of visit in the main and control groups, the study was 0, 3, 6, and 11 months. 4 visit interval. So transplantation result is the remission of nephrotic syndrome, trophic skin and maculous manifestation, decreased activity of the autoimmune process and reducing the dose of immunosuppression to minimum. Affection mark is kidney biopsy, pro-inflammatory cytokines, antibodies, and glomerular filtration rate. Stem cell transplantation consists 3 stages, 3 main stages. First is myeloexcision from the ileum, and after punction of skin, about 220 milliliter of bone marrow is aspirated. After this stage, the bone marrow mixture is delivered to the laboratory of stem cells by technology, where the mesenchymal fraction of stem cells is isolated and pre-cultivated for 7 to 10 days. This picture shows stem cell sampling and biotechnology phases. After bone marrow sampling, we isolate all cells and bone marrow cells cultivation in special condition with mixed medium and growth factors. Next, we take electron and binocular microscope examination, test for sterility of cells and cell count of medical calculation. Determination of phenotypic composition, in particular the percentage of CD73 plus cells, bearing the bone marrow cells as the marker, cultivation for 3-4 weeks, and obtaining MSC about 19% mononuclear from 80 120 million cells. The next stage is transplantation at the National Scientific Medical Center. MSC transplantation is carried out systematically at a rate of 50 milliliters per hour. The transfusion time is 2.53 hours. The multidisciplinarity of the procedure is 3 months. If the criteria of efficiency, one of criteria is efficiency, my criteria is result of biopsy fitness of patients before and after MSC transplantation. First, we can see before the introduction of stem cells, we can see glomerular cells proliferation, expansion, and interposition of the mesenchyme. Membranous changes in the form of local gelanosis in the sclerosis of vascular groups, tubular damage, and fibrosis of the interstitium. It's before transplantation. After 3 months of transplantation, we can see decreased proliferation of glomerular cells and slight decrease on the amount of sclerosis of vascular groups, a reduction of the interstitial fibrosis. Is staining with gematoxylin and eosin. Next. Next, the result, picture shows the results pathomorphological research. After 3 months transplantation, we can see decrease in the intensity of staining of the tubular interstitial zone. It's EGA manifestation 200. Second staining, it's before treatment and after treatment, post-treatment. Decreased inflammation as indicated by the intensity of glomerular and interstitium staining. This is result of Jones staining before transplantation and after 3 months of transplantation, a slight decrease of the intensity of growth staining of the glomerular and interstitium. Second picture is this chemical staining path reaction. After transplantation, we can see reduction of the local and segmental glomerular sclerosis. Next, demonstrated before, also before and after transplantation, staining with gematoxylin and eosin and mesotrechrome. According to the staining with gematoxylin and eosin, we can see decrease in the degree of the kyanosis and sclerosis in the glomeruli and dystrophy and tubulus. After treatment, according to the mesotrechrome, we can see reduction of the intensity of the blue staining, the connective tissue in the glomeruli and interstitium. After the treatment in the kidney tissue, there is a positive trend, which is expressed by histological examination staining in a decrease in the degree of kyanosis and sclerosis in the glomeruli and dystrophy in the tubulus. This chemical examination is of mesotrechrome showed a decrease in the intensity of the blue staining of the connective tissue in the glomeruli and interstitium. Jones silver staining for elastic fibrosis showed a slight increase in the intensity of growth staining. This picture shows the functional state of the kidneys, dynamics of daily proteinuria in the main and control group. We can see the drop, daily proteinuria in the main group, and second picture is dynamics of glomerular filtration rate of the main and control group. We can see positive trend in picture 1 and picture 2 inform changes of dynamic of glomerular filtration compare control group and main group. Also, the microcriteria for the efficiency of our procedure is functional state of the kidney. We also investigate the cytokines and level of TNF alpha and interleukin 10. Decrease in the level of anti-inflammatory cytokines was also observed. We can see this trend in the picture 10 and 9. TNF alpha decreased from 2 to 0.92, and interleukin 10 from 6 to 2. This is the decrease in proteinuria. It's a framework of lupus nephritis decreased from 6 to 1. Thus, the studies showed a significant decrease in the activity of autoimmune process in the kidneys. Positive morphological and functional changes in the kidneys after bone marrow MSC transplantation have a positive effect on the overall clinical status of patient. Clinical efficacy was not in all patients by the first month after, and over the 6 months it continued to increase. It's allowed us to reduce the dose of methylprednisolone and immunosuppressants in 85% of the studied patient of the main group. Thus, transplantation of autological bone marrow MSC for treatment systemic lupus erythematosus, according to the clinical and laboratory experimental data, was significantly more effective than traditional therapy. Showed a significant decrease in the level of immunocompetent cells, blood cytokines, and improved clinical parameters. Morphological assessment of kidney as noted earlier after transplantation of MSC obtained from bone marrow showed a decrease in the immune inflammatory process in the vascular loops and glomeruli. Bone marrow MSC transplantation has advantages over standard therapy since it improves the natural source of the disease, is safe, and allows living symptom of autoimmune aggression, which positively affect the function of target organ in systemic lupus erythematosus. The pharmacoeconomic efficiency of the use of MSC was noted since about 80% of patients with systemic lupus erythematosus, the dose of methylprednisolone and immunosuppressant were reduced. Thank you very much.
Please. Please. Please, open.
Yes, could I better understand? So, where do you think they go after you've injected them your MSCs? Do you know whether they go to the kidney, or do you think they'll probably end up in the lung or potentially the liver? So, when you did your biopsy, did you see an MSC in there? I mean, it'd be hard to find them because they're autologous, aren't they? But have you labeled them? Do you know where they go?
Thank you for your question. This presentation demonstrated a clinical practice. We use stem cells for treatment of
Вопрос был, знаете, какой идут СМИ, когда откалывают. И тут, может быть, селезенку или один с почтой. Вы как-то имеете, или вы смотрели?
Системное введение идут, Настя, по закону именно первый печень, орган, легкий, и по тем местам, где команда СОС, опять это МСК. То есть орган мишень, в данном случае СКГ, автономный процесс поражения почки. Вот мы показываем эффект.
Но они там есть, да?
Да, они есть. Безусловно. Они идут по закону, идут туда, где есть патология. Экспериментально это у нас 2000 года у нас в эксперименте все прошло. Наша клиника более 20 лет занимается точными технологиями. И мы прошли 3 научно-технических программ. Значит, потом скажем, научно-технических программ по заказу МО и Минздрава. Соответственно, мы перешли на уже экспериментальный, научный, практический, клинический, показывали. Это наш опыт, он докладывался в Италии в прошлом году с успехом на Евро-дерматологическом конгрессе. ССД есть.
So maybe kind of translate the answer, yes, the cells do grow in them.
But this is not in this experiments. They showed
So
the
they-
no,
no, it's
yes,
long
no,
time
no,
ago.
in this experiment, just guess that cells go to the place of the injury inflammation.
But the thing about lupus is that it's a widespread autoimmune disease that of course affects the kidney quite severely, but it affects many other organs as well. And you've shown pictures, of course, of the skin. You know, this is, I just wondered, so I do have some very specific questions, but I didn't understand the premise, the hypothesis for why anybody would think this would work. It's a strange, so is it that somehow the MSCs are just like an anti-inflammatory drug, or why do this?
Какой вопрос у него? Вопрос такой, что какая гипотеза у вас под этим экспериментом? То есть вы здесь как антиинфламмейшн. И какая гипотеза, почему вы видите, что липус болезнь, например, системная, то не говорю, какая гипотеза была исследована?
Прежде всего, ну вот как прозвучали до нас доклады про мезонхимальные стволовые клетки. Поскольку мезонхимальные стволовые клетки обладают противовоспалительным, иммунорегуляторным, да, иммуномодулирующим эффектом, а системная красная волчанка, системная склеродермия, прежде всего, это аутоиммунные заболевания. И, конечно, гипотеза в том, что МСК, выделяя вот эти, оказывая иммунорегуляторный эффект, противовоспалительный, выделяя различные факторы, супрессирующие аутоиммунный процесс в очаге. Вот, в данном случае почки и губы. Да, конечно, это системное заболевание. Выделяет трансформирующий фактор роста бета, прежде всего, на сегодняшний день самый известный, и модулирующий плеотропный иммунорегулятор. Конечно, супрессирует именно аутоиммунное воспаление. Системно и вместно. Вот, вместно мы не вводили, системно, конечно, он оказывает лучший эффект.
Hypothesis since lupus is autoimmune disease. First of all, consider even the regulatory hospitals of stem cells. So because expression of
Кроме того, поскольку имеет место аутоиммунное воспаление, а именно МСК, как доказано, мы и сами доказали, и как вот в обзорах литературы, обладает антиапоптотическим эффектом, когда экспрессия этих факторов апоптоза высока. При введении мы еще указали вот такие, как антиапоптотические факторы, как аспаза, 9, вот тройка, антиапоптотические тоже мы изучали.
this is autoimmune.
It's sort of, and then specific questions, so I didn't understand the controls. So were there a set of when you're comparing the kidney, were there some individuals who received those stem cells before and after? I think
It was my question about ask and translate. So, вопрос хотел задать, что было контролем. Показывайте контроль, что было
контроль. У нас контрольная группа была, их очень много. Такие больные у нас лечатся, конечно, это очень большая группа. Вот. Мы контрольной группе также одинаково принимаем только метилпреднизолон и ВИС, это высокодозировочная сильная терапия. Преднизолон, и у них, конечно, результаты качественно даже.
У вас были люди, получали
стандартные, которые половина.
How then we sit on standard treatment and how was the standard treatment plus.
So the improvement that was presented was against the stem cells and no stem cells, and then the second one.
Так, чтобы показать контроль, у нас была группа, которая сначала только кортикоиды, да, а улучшила. Улучшение кожи и часто кстати, можете знаете, там, первая правильно показывала, ну, еще хорошо. Следующий. The question is regarding to setting power of that data, because what I see from there, for TNF, you don't have statistics. Yeah, but if you have TNF, what you create to say that you decrease from 2 to 4, and you show P 0. I don't see it. What's statistical power? Just to tell us.
Мы еще насчет клинического эффекта.
Здесь получается, ну, как бы, the two groups statistically
То есть статистическое улучшение достоверное вот именно
Вот я спрашиваю достоверность. Там не показательная графика. Вот, например, я сразу говорю, на третьей группе я не вижу статистического, если показательная ваша, да? На второй я тоже не вижу, скорее всего. На первой может быть, поэтому я спрашиваю, где там статистика.
Данные представлены.
Да, во-первых, что это у нас? Это стандарт эррор, мини стандарт эррор. И потом какие методы вы обрабатывали? Данные.
Yeah, I think it's from the various changes between the first visit and the subsequent visits. But, I think what would be useful is thinking about it being autoimmune. It might be sensible. And I wondered, have you got any ideas about perhaps measuring levels of autoantibodies that occur in the circulation? Do we know whether or not there was a reduction in the autoimmune? The IgG would be a simple one, I guess.
Понятно. Кроме того, можете перевести, да? Можем как бы ремарка какая. У нас группа контроля, и основная группа, да, болит одинаково СТВ. Основное преимущество это женщины. Вот. И у нас есть опыт такой, когда 10 женщин, которые замужем в протяжении 10 лет, да, им обычно запрещают рожать, отчеты, клинические вот эти дела. Вот. Но поскольку эти улучшились лабораторные вот эти показатели после трансплантации мезенхимальных стволовых клеток, у нас 10 случаев, когда женщины беременели или успешно рожали. Когда как контрольный эффект этого нет.
So direct answer, but it says that in that case, they have women patients who did not give a birth to get pregnant.
Да, это системно как раз.
And after treatment, some of them became pregnant and gave a consistent birth to a child.
I think having a reduction in antibiotics would be something that everybody would understand as well. It'd be worthwhile investigating that. That would be so. While TNF is interesting, I think it's autoimmune. You show a reduction in antibiotics. This would be something.
Your recommendation, right?
Yeah, I think so. Something that we need to measure.
Через файлы вы как бы отзывы показали, вам неплохо было бы еще померить антитела.
Это все мире. Механизмы далеко там. Механизмы еще далеки от разрешения. Мы в дальнейшем надо это. Анна этому не делает.
Quite a discussion, so we told a little bit why you all came here, right? Presenting of discuss the results. So it's my great pleasure to present the last speaker of this session today, Dr. Alexey Lindu, who came from People's Friendship University in Moscow, Russia. He's a head of the research and education working on stem cells for long and collaborating with us with Neroclass, and also for quite some time. So the title of this talk is Legal Regulation of Cell Preparations in the Eurasian Economic Union. So we are really relate. Well, this maybe asking what you would describe and then questions for the discussion during this presentation.
Hello, dear colleagues. Thank you for the opportunity to be here. Let me present a report on the law regulation of cell products in Eurasian Economic Union. The report includes a classification of cell products, the products where the culture itself is the active pharmaceutical substance. The report also includes a comparison of the regulation of European Union and the Eurasian Economic Union in the field of approval of cell products. Finally, I will give an example of my own development of a cell product, which was supported by a grant from the Minister of Industry and Trade to be approved in accordance with these rules. Okay, today there are more than 45 cell products are approved in the world. There are drugs based on different cells, chondrocyte 11 drugs, skin cells, keratinocytes, and skin fibroblasts, 9 products, genetically modified cells, 8 products, mesenchymal stromal cells, 7 products, and other cell types. Which cell products are expected to be registered this year? You can see on the slide that the cell and gene products are being actively registered for introduction into clinical practice. It is also worth saying that before, in the early 2000s, the main indication were the restoration or regeneration of skin and skeletal tissues. And now the focus has shifted to the oncological disease. In general, the trend towards the increase in cell and gene drugs on the market confirmed by the statement from FDA, which expects to register 25-30 such drugs annually in the coming years. The principal stage of the development and registration of cells are the same and stated in the regulatory frameworks for various countries. In general, this stage corresponds to those of pharmaceutical products. Currently, there are no registered drugs in Eurasian Economic Union, but starting from 2025, all medications including cell products must be registered within this framework. It should be especially noted when moving to clinical trials, the cell must be GMP grade. Within the framework of Eurasian Union, the following requirements for medicines have been adopted. These are GLP, GCP, GMP, GTP, and general requirements for pharmaceutical inspections and also the Union Pharmacopoeia. Thus, a single market for medicines is being formed. To what extent do these GXP rules comply with the same rules of the European Union? Almost 100%. In fact, it's practically a translation of European documents. The difference exists only in the field of registration model. The following classification has been adopted. Cell preparations are biological drugs and are divided into 3 types. Gene therapy medicinal products, somatic cell therapy medicinal products, and tissue engineering products. Features of the regulation of cellular products are the following principles. decentralization of regulatory bodies in the Union, 5 regulators. There are 2 approval mechanisms, mutual recognition procedure and decentralized procedure. Relationship with national regulation in the field of circulation of human tissues. What is the relationship between supranational regulation of cell preparations with national healthcare regulation? The fact is that the cell preparations have the following feature. Since the initial biomaterial is human donor material, it's necessary to take into account the national regulatory framework that regulates the handling of different types of cells and tissues. It turns out that different cells are regulated by different laws. I will give you an example in Russian. The fact is that different types of cells are regulated by different laws. For example, bone marrow is the law on transplantation. Hematopoietic stem cells is under the law on blood. Thus, when developing cell preparations, these features must be taken into account. Okay. Our biomedical cell product is a product of cell therapy. It's suspension of autologous MSCs from bone marrow in physiological saline for external use. The safety and efficacy of MSCs in the treatment of patients with the neuropathic form of diabetic foot ulcers was demonstrated in pilot study. This slide shows the first patients with diabetes mellitus, diabetic foot ulcers with non-healing wound of about 4 square centimeters. We applied the cells to the entire surface of the wound, but epithelialization starts from the edge. So, the day 10 after the application of the cells, we noted a significant reduction of the wound size to 0.7 square centimeters. By day 18, the wound was completely closed. This result inspired us, and we began to recruit patients with much larger wounds. Patients with large ulcers up to 40 square centimeters also demonstrated good wound healing dynamics. Also, the feature shows the rate of epithelialization in the group before and after the application of a single dose of MSCs. The axis Y shows the average level of epithelialization in square centimeters per day. The average rate 2 weeks before the use of cell product was 0.9 centimeters per day. This rate was approximately the same for ulcers. So the rate of epithelialization was not different for patients. A single application of biomedical cell product increased the average epithelialization rate by 8 times. This result was statistically significant. The epithelialization rate decreases with time and is 5 times for 21 days. In general, the last column shows the average speed of epithelialization to complete healing. There are the results for all patients with different sizes of ulcers, which were published in the Journal of International Society for Cellular Therapy. Thank you for your attention.
Thank you so much for this brilliant presentation. Any questions?
What about the legislation? Because we heard that recently, like not recently, around 3 years ago, there was some kind of a law in Russian Federation that legislate the use of stem cells and overall the cell products. So how you manage with it, and like do you obey the rules? How the therapy goes along with legislation?
Yes, you're absolutely right. Now in Russia there are two regulations about cell preparation circulation. The first one, national regulation, 180 law number, and now we have a process of combining these two regulations. So biomedical cell products will be the ATMP according to Eurasian European Union. ATMP, advanced cell therapy, for instance.
Okay, but who's going to control what the hospitals are doing? I mean, the process of preparation, how it's going to be controlled, how you can allow one treatment and, for example, decline another one, like based on the, for example, markers, or how does it work now?
Okay. That's why we have two regulators, one by Ministry of Health and another one in the field of regulation of Ministry of the Trade. So in the future, I think in a few years, we will have only one regulator with special inspectorate for GMP manufacturing places, and it will be Ministry of Trade in Russia and in other countries, in other regulators, applied for regulators for all Eurasian Union.
Thank you.
So you think, just in the continuation of this question, your product, the usual one for results, so how long it will take till it will come to clinic?
Thank you for this question. It's a really long way, and now we have only formulated GMP design of our product, and this year we will start a preclinical trial, and I think that the approval will be by 2027, I guess. For this
So in 4 years from now.
Yes.
Maybe I can tell something about Europe, because it is reflecting into Europe. The problem is that what you do, and then what you all develop in the hospital, that is sometimes allowed because it's university, you develop new approaches. But as soon as you want to go to a product range, not a transplantation, but a product range, then the regulation, what Alexius is telling, is enormously time consuming and regulatory consuming. Because it's not only that you have to build the product, you have also installed the quality controls, the way that you demonstrate outside the body what the product does. And that is quite a challenge, I think. For a simple product, this is quite a challenge.
Thank you for this.
And in Europe, there's a hospital exemption rule, and I think that that's the same maybe in Russia, that you do it as soon as you do it on small numbers in the hospital, completely controlled by the responsible physician. Then in Europe, regulatory do this. But it is still not allowed, but it's a gray zone. Otherwise, you cannot develop. If university cannot develop, nothing comes on the market.
Thank you so much.
Okay, yeah, colleagues, it looks like we covered the main items of our agenda. That's all for this section, and thank you for all of you for your attending. And we welcome you to coffee break. Feel well.
Yeah, but
Thank you.
let's take a look here that. Now it's
Okay.
history. And like to suggest, and I hope you don't mind to be back at the same time. answering, so I will help him. He has to come back to answer the questions. One day.
Yes, because this approach would be very interesting to our pediatricians. Also, we still have problems. Everybody has problems, premature birth, and how to make our babies just provide normal life in the future.
One of the things maybe for you when you were working with sheep, you think I'm going to develop an animal facility also for sheep. He is enormously experienced with pulmonary development in premature sheep models. He has a lot of knowledge on that. If you want to do it, something in that direction, I'm happily to connect you to him, because he is in Australia also doing that.
For sheep.
Yeah. He can tell you everything about sheep development, pulmonary development.
It's very important for Australia to see sheep.
I only know that they taste.
Why I know that the clothes are good to get done.
No, but I connect you with him. He can help you with that.
Well, it's not like my particular interest. I just now know people who could be interested in it. Maybe you can start production cells and once. Just so.
Do you think with pediatricians,
Yes. For example, we can finally organize a well-done facility for cell production. I think a lot of doctors would be interested.
Okay, dear colleagues, our next speaker from Laboratory of Bioengineering and Regenerative Medicine, National Laboratory Astana, Kazakhstan, Dr. Yulia Safarova. Miss Yulia.
Hey, one word about here. This 90% of effort that all that event took place, organization, online connection, stuff like this, basically it's from.
yes.
Thank you a lot. So I'm not sure whether I should use microphone, потому что нас много, мне кажется, или лучше использовать. Okay, thank you for introductory word, and I'm really honored to present at this kind of event. It's a small workshop, but it's really informative and important, especially for me. And today I'm going to talk about stem cell therapy for bone fracture regeneration in a small animal model. And we talked a lot today about the mesenchymal stem cells, and our lab is also doing this research. So I will show you and describe several of our projects and several directions of how we manipulate mesenchymal stem cells. And so I won't stop that mesenchymal stem cells are largely extensively used nowadays in stem cell therapy for manipulating for treating any diseases. So but main issues of stem cell therapy that we encounter is a loss of homing receptors and their survival. So this is the main approach. So how we can enhance the efficacy of stem cells, particularly stem cell therapy. So we can entrap cells into scaffolds to give them more stability, to give them some room to grow and to differentiate. And this is used in orthopedic research a lot. We can manipulate them genetically, but this has its pros and cons, because the therapy that's going to be genetically engineered places a lot of fear to all the patients and to society, and to even do the research in this area. And another one approach is the surface modification of cells. And so this is how we come to the idea about modifying the surface of stem cell to give this a targeting potential. So the cell will have the affinity for something, and we use this approach in bone regeneration
So what we did, we collaborate with another institution and we make the polymer that has bisphosphonate on one end and NHS group on another end. So this is the bisphosphonate group and it has high affinity to bone tissue. And the NHS group, it has affinity for membrane proteins on the cell membrane. So we would attach this three cells with this polymer to make them be stick to the cell. And so why use bisphosphonate? Because the affinity to bone tissue is already proved and they has high affinity. And besides, they also can inhibit the cytoplastic activity by itself, because they interact with farnesyl pyrophosphatase, the enzyme that actively participates in osteoclastogenesis. And so our hypothesis was looking quite similar to that. So we had mesenchymal stem cells and we have this bone targeting polymer that has NHS group to bind to the cell and bisphosphonate group to seek for the bone. And so that was done in collaboration with Carnegie Mellon University. So we've done it by atomic transfer radical polymerization. The whole polymer was engineered. So it has a lot of pros, the positive sides. So in polymer engineering, it gives you the functional, so you can introduce the functional group and you can actually say how many molecules do you have on the polymer of these functional groups, and that's really great. So we had a polymer that has NHS group, and then we add the bisphosphonate group, and we also had the polymer that did not have bisphosphonate, it did not have NHS group, to see the control and to see how good they are coated, how good they are targeting to bisphosphonate. And here is the cell binding. So we did also a lot of in vitro studies, and I will skip them a bit, but we showed the polymer stably attached to mesenchymal stem cell by introducing the pleuristin molecule on this polymer. So we can visualize them under the microscope. And we also tried to see the affinity to the bone fragments in vitro, and we proved the affinity of polymer towards the bone chips. And we checked the cytotoxic effect by luminescent cell viability test. So cells are not toxic to, polymer is not toxic to mesenchymal stem cells, and the polymer does not affect osteogenic differentiation. So this is another study that was done to prove its anti-osteoclastic activity, and we kind of had results in it. So, and then we transitioned to in vivo studies. And so the approach was to test in osteoporotic model. So during bone regeneration in osteoporosis. So we induced osteoporosis in mice by bilateral ovarectomy, and we monitored the bone density with IVIS, the in vivo imaging system that we have. And then we performed the ulnar fracture model. So this model was actually transferred from one research that used rabbits, but we thought that it would be beneficial, more preferable in rat, since the adjacent fracture serves as a spleen and it provides weight-bearing support. So we can see the bone staying into the place, the ulnar one, the one we are breaking. And the radial adjacent radial bone gives it the support. So we can monitor how this, how this bit here is regenerating the fracture. And this is was experiment outline. We've done the OBX model, and then we've done the fracture and the treatment. And of course, we had 0 week, 4 week, and we tried to monitor animals down to 24 weeks because osteoporotic fracture really hard to heal. We performed bone marrow or bone mineral density assessment, and after all, we performed histological studies. And here are the control groups. So we injected only PBS, then there was only the polymer dissolved in PBS, only mesenchymal stem cells in PBS, and polymer modified mesenchymal stem cells in PBS. And this is about analysis of bone mineral density after 4 weeks of treatment, and we monitored significantly important increase in mesenchymal stem cell plus polymer group. And so did the increase a bit decreased. I mean, it became 21%, but still it's good, and it remained permanent for 24-week duration. So 24-week period.
Yulia, can you show that in untreated animals, bone density
Okay.
decreased?
Yes. So while the mesenchymal stem cell therapy gave some kind of a increase in the beginning of the treatment compared to the negative control, during 24-week time, when the rats were developing a osteoporotic condition, and we had this decrease even more. So that was like kind of a negative effect of mesenchymal stem cell therapy in long-term perspective, and polymer effect in long-term perspective. But this difference was not significantly important. That's why not mentioned. And here is a representative micro-CT images after 2 hours of fracture, 4 hours of fracture. So we can see the fracture is still there. Then we perform histological analysis, and I won't go deep into that because we have a second approach. And so histological analysis was also positive. We have ossification and bone tissue formation and complete healing almost in modified mesenchymal stem cell and polymer group. So we're also trying to monitor this fate of cells that go into the fracture to see whether there's an effect of the cell itself or the cytokine they're producing. So that's a eternal question, what we all are questioning, like, and asking and trying to investigate. So we transfected cells with luciferase lentiviral producing elements, and then injected the cells into the animal, and here we go. So we had this mesenchymal stem cell group and mesenchymal stem cell group with a polymer. We were able to monitor them for 1 week, and at week 2 we had no signal. And it's interesting how about these drops, I think we can discuss during the question time, because when you inject cells, they produce almost the same signal, but after 1 week in a group of mesenchymal stem cell and polymer, the signal goes really low, and we hypothesize, like we would like to hypothesize, that this is due to migration of the cells inside the bone tissue, because the IVIS is not able to perceive the signal from under the bone, like luminescent signal. That would be a really good explanation, but it's still a hypothesis. And another strategy that we use is enhance the mesenchymal stem cells, because transplantation of mesenchymal stem cell is usually required for older patients, and the proliferative activity, migratory potential, and like stem cells, they do age, and to rejuvenate cells, we this is the idea of getting the old cell manipulated down to the young cell parameters and increase their proliferative activity, increase their migratory potential. That would be great. And so this is how we come to idea about the changing the quality of transplanted cells. So and this is the research of Dr. Baujan, where I was also participating. So the project was that focusing on CDC42 is one of the key regulator of cell senescence. So we inhibited the CDC42. There were actually some publications, a lot of publications, and we showed it in vitro before that when you inhibit the CDC42 and downregulate this mechanism, you will have the positive effect in vitro. And so there was a paper published in Biogerontology. So our hypothesis was that this inhibition of CDC42 activity may improve the therapeutic efficacy of bone fracture treatment. What we actually done, so we performed the in vitro studies, and that included the differentiation of the blast, angiospastic activity, migration activity. So in all these cells, we inhibited CDC42 with a pharmacological agent taxine, that's well-known inhibitor of CDC42. And then we transferred to in vivo studies where we were injecting and the stem cells inhibited the CD42 into the ulnar fracture, and so this is what we got. We had at 16 weeks showed persistent increase in group that were injected with mesenchymal stem cells with casein. And so, yeah. Do I still have time? How much is it?
Also,
Yeah.
also did silencing.
Oh, we were not showing this. This slide, it has this, like we silence that was performed the silencing of CDC42 with siRNA, and so with the siRNA we were doing from Life Technologies, and it also had really good results compared to casein, even better one. So silence of CDC42 resulted in almost twice increase in bone density. Yes, so this is just a short explanation about the inhibition of CDC42, a lot of in vitro results and so this is how we measured the migration potential. So we've done the Translational analysis, we also done Kureysh assay, and what else? We've done the F-actin staining. So F-actin is really important in migration and cell dynamics. And here was the in vivo assessment. And so in vivo assessment that include only casein. So we had this increase in 1 month. And I would like to thank National Laboratory Dr. Sultana Askarova, who was my PhD supervisor. So the first part of the project was my PhD research, and Dr. Baujanov, the second part belongs to him. And yeah, and that's great that you came here. We are really lucky to have this kind of professors visiting the Zbark University and National Laboratory in particular. Thank you. I do expect a lot of questions. Yes, go ahead.
So I probably missed it actually. So
Yeah.
it's the first question. When you put the polymer onto the cells, how did you assess this very simple question? Did you assess viability of the cells downstream? Did it change?
Yes.
And yeah, so
Here is the viability. So we've done it with luminescent cell viability assay from Promiga. I do really like this kind of assays. They have like really easy.
How long did you wait?
So that was short term for 4 hours and long term for 72 hours.
So basically, not as long as, so if these cells were to, if you thought they might proliferate, or what you looked at is when you said your signals disappeared after 1 week. Was it after 1 week that you lost the signal?
Yes.
So you don't know whether or not the cells could have lived for 1 week.
Well, that was done for 72 hours because 72 hours is the maximum for them to, like, the time that it takes them to travel down to the bone tissue. Hypothetically, but one week.
Just go back to that survival.
Yeah.
So just that, is that death? What are we looking at here? Is that death? So basically, are they dying in 72 hours? There's no difference in death.
No, they are not dying. So that comes 4 hours. So they start double at 24 hours. What I was showing here, so you have 1 hour, 2 hour, 4 hour. This is the short-term toxicity, and here comes the kind of long-term 1 day, 2 day 2, day 3. So they start double, and the
what so in fact they're getting to multiplied cells, so they're proliferating. It's not affecting their proliferation.
Yes, even.
So then what happens to the polymer on the cells every time they proliferate? What happens to the amount of polymer? I guess it gets depleted.
We did it. Remember, we did experiments. We saw the internalization of the polymer.
Internalization of the polymer. How long? So it takes 4 hours for polymer to get internalized inside the cell. And so and because the polymer is new, we don't know about the pharmacodynamics and how the cells what's happening inside the cells with the polymer. And so what we do pretend that it loses the fluorescent group after 4 hours, because when we were doing this kind of experiment and monitoring how long the signal stays on the cell, we can only rely on the fluorescent group that's inside the structure of the polymer. And it's fade away in
12 hours, 12 hours.
12
12
hours, after 12 hours.
Yes, no.
Yeah, so we lose the signal, but we are not sure, are we losing it due to fluorescent group or just fluorescent group with the polymer gets degraded?
I mean, first of all, I think I should say that it's all really elegant and fabulous, really good work. And just to make it clear, you didn't put the polymer on when you manipulated the mesenchymal stem cells in the second part of your talk. That wasn't plus or minus polymer, so you haven't combined the two approaches yet. Is that right?
No, we did not combine the two approaches because two main variables.
Okay.
So approach, so what do you mean?
CDC42, you mean CDC42
Yeah,
and
exactly.
and polymer.
These separate projects, they did with separate things
in a very time frame.
So your polymer, it wasn't clear to me, it's basically the linker arm.
Yeah.
How long is it? And the reason I'm asking this question is because we've decorated cells with some ligand targeted. We basically developed some modified surface of cells with some sugar. And it worked very well on liposomes, and we got targeted drug delivery. But as soon as we push it onto cells, what we think happened is that the label disappeared into the forest of proteins on the outside of the cell. So we think we took something that's very small and we put it, it's a little bit like, say, putting a small yurt in the middle of New York City. Basically, it was obscured by everything else. I just wondered whether you discovered whether the length of the linker makes a difference.
Yeah, so the weight of the polymer is 8 kDa, and right, and no, we did not do it because we should probably do it. So explore the cell membrane, how do they attach, or what we did, we actually measured the polymer molecules, how many polymer molecules is needed for cell. That's how we calculated the concentrations for the polymer to cover the cell. So we did this back in at the beginning, but we did not made it again.
Okay, it might be something that I think that may be why you're being so successful, because you've got that long linker on it. But,
yeah. Thank you. Thank you. Any questions?
Yeah, I have a question. Thank you for your great presentation and for great methodological design for this experiment. The question is, you know that initial situation, the gold standard for bone fracture with large bones is outer transplantation of bone fragment from another bones. And the second choice for surgeons is osteoclastic materials, especially allogeneic bones. What do you think? Maybe it would be interesting to compare the effectiveness between your tissue engineering bone and clinical methods.
Well, yes. So for critical size defect, I'm as a researcher can say that you would like to have something that will support your cells. So there's allogenic bone and stuff like that. But for some condition that cannot be engineered with this construct, like the stem cell therapy is a good alternative. Exactly.
Because I can answer this question. So what's basically behind this project? When you have a general fracture, normal people are not to heal anyway. Because some people they just wait. Here we're talking about osteoporotic fractures of all people. Wherever they have fracture, they never heal. And it's first. And second, you cannot put, that prosthesis to that bone because it's very big. And we talk to our orthopedics, they just cannot do it because there's no way for them to put that, whether it's artificial bone, wherever it's bone transplant or wherever it is.
Metal construct. They are saying
the surrounding bone just cannot keep it. This is one of the problems.
I think that we're doing this in OBX life.
in overtime.
Yeah.
It was done.
Oh, sure, those were.
Yeah, it was.
It's not just bomb. It's a whole idea behind the project.
Oh, yes.
It's another side she didn't mention. This neurodegenerative also has osteoclastic. I mentioned it and many well as well. We show that Alzheimer's reported cells they reduce osteoclastic activity of osteoclasts. This is another therapeutic. We see there's two ways. One we make a proof of stem cells, and second we reduce osteoclastic, reduce the reduction of bone. So this was all behind that.
That's a kind of secondary effect.
This osteoclastic images we can.
We can discuss like the osteoclastic moments later because osteoclastic, reduced osteoclastic activity goes to the loop that will eventually decrease the osteoclastic activity. So I would like to talk about this during the coffee break.
Okay, thank you.
Thank you.
Okay, yes, I can thank Julia for its presentation and once again.
Thank you.
And I would like to ask the audience, so we have optional break indicated in the program. So it's up to you whether you make a short break or we just go straight.
Walking still there.
Walking still there.
Makes sense, I think.
Yeah, well, so.
No, but it's the fear and the coffee break for work.
No, that's a practical consideration, so.
Nobody from here. Yes, from this order, nobody executive.
Alright, so, yes, coffee break, yes, coffee break, 5 minutes, yeah, okay, that's the breaking point, alright.
I did so. Now, first, the guys just go for it. I have to try to teach him more language to this, and I only know yes, so
Сам концепт. Екатерина, очень приятно. Конечно. Да, он мы чуть-чуть опаздываем, поэтому будет сейчас Дэниел, потом Евгений Пономарев, а потом уже вы. Психология. Это ваше, да? Да. Психология. Вот, PDF.
You actually so did you insert yourself?
Просто... нет, все нормально. Да, кликер, главное, чтобы работал. Знаете, вот, ну, если его раскрыть, он должен работать. Как это делать? Запускается PDF. Вот, полностью, да-да-да. Ну, у нас, по-моему, не... Нет, должен. Да, давайте я накрою направо и до конца, и там направо.
But it's not a great like a picture I just different thing.
Apologize for going out.
No, that's fine.
That was only mistake more than I think that take it on the cover. Yes, tonight, yes.
I would like to talk to you on the neuro self.
Okay, hold on, be free. I'm here, but I apologize because I was interested in, but that's my stupid colleague.
That's fine, no problem.
Was so disturbing. I don't do that, but.
That's okay, no problem.
So, what do you think? How are you? It's still functioning, they have a very.
Yes, yeah, but Boris was calling you, very me.
But
Het is wel organized.
Okay, so we promised 5 minutes. 5 minutes ago. Unless they will need more time to finish this copy. So I have a pleasure to introduce again Professor Daniel Anthony from the University of Oxford, Department of Oncology, where he's a full professor and head of a deep laboratory, working in neuropharmacology, surgery, neuroscience, working with us.
Thank you. All right. So in view of all the presentations that we've gone today, I was going to talk to you about lots of interesting things we're doing, manipulating the microbiome and changing behaviors. But then it became clear that actually, I think, given the reason we're here as well, that discussion and presentation of some, it's going to be a short presentation, just showing you some new results that we've got that Hans alluded to, where we've injected some of the neurocells into the base of some TBI lesions, and we had a look to see what sort of recovery we were going to get. And I should say at this moment that I'm a bit of a skeptic when it comes to any of these experiments. And I thought that what I'd start off by doing, take a little bit of time of reviewing some of the unique problems associated with trying to treat neurodegenerative diseases or injury. So this is about TBI, but and to think about what difficulties present themselves and why it's an issue, really. And I think that it's worth reminding ourselves what's different about the brain that makes it so problematic. So the first thing to remind everybody is just to try and avoid the wheelchair altogether. The cleverest thing you can do is not do an awful lot of surfing. Try not to, I know I'm talking to the wrong people here because horse riding is a fairly serious source of spinal injuries. But wear your helmet, wear lots of padding, big suit out here, I think is probably there, but avoid the head injury or the spinal injury as best you can. Because fundamentally, the reason we need these wheelchairs is that we just don't regenerate, and that in the central nervous system. Now, you will hear so many people talking about the way in which the brain is able to, where we have all these interesting sort of progenitor cells that can give rise, the hippocampus, for example, to new neurons. And to those people, I always say, if it were the case that the brain was very good at generating new neurons, that adult neurogenesis was an important thing and a significant thing, we wouldn't need wheelchairs. It's very simple. And I think the best thing that we can, that I often use to illustrate why we don't regenerate particularly, is you have to imagine the complex machinery of the brain. Now, I don't know how many neurons you have, but I have about 100 billion neurons. Now, that's more than I can count. And if I started tomorrow, I would still be here in a little while. So you imagine all these neurons talking to each other, and then you decide to damage a bit of the spinal cord or the brain, and you pull out or effectively destroy all the connections, and then you ask, hey, let's just rewire the brain, and it's going to be fine. So the starting point was to say, was discoveries such as BDNF, for example, or NGF. NGF is a really good example. NGF, we know, in a tissue culture dish, causes neurons to start sprouting. That's great. So I tell you what, now we know that it'll do that. Let's stick it into the spinal cord and see what happens. And to individuals who initially injected with this growth factor into their spinal cord, they developed some really horrendous pain syndromes. It transpired and it generated a very interesting field in neuroscience, understanding the contribution of TrkA and TrkB receptors to pain syndrome. So fundamentally, we discovered that all the things that we thought would be really clever, let's put in a growth factor and we can regenerate the central nervous system, doesn't work. So we're left wondering, first of all, so rewiring the brain is a complex thing, but that hasn't stopped people trying to think about how we could just put some cells in and somehow it would be sufficient to encourage enough regeneration to actually result in some sort of positive functional outcome. Now, I'm going to just review very quickly some of the work that was done by a good friend of mine, Martin Schwab and Lisa Schnell, very many years ago. And they discovered a no-go and then subsequently the no-go receptor. So one of the questions that was always asked is, why physically don't axons basically regenerate? Why don't they grow and find new targets in the central nervous system? It was Martin who discovered no-go. And they found an antibody that basically encouraged neurons to sprout and for axons to grow and try and cross lesions. And it transpires an interaction with no-go receptor, no-go on the oligodendrocyte, effectively causes growth cone collapse. So intrinsically, the central nervous system is designed not to regenerate. So you can overcome this. So we take an injured neuron, it degenerates, and then we imagine that what's going to happen is somehow the neuron has either got to try and bridge the gap, cross the scar tissue, and then grow and find its target. Or conceivably, actually, maybe the cleverest thing is all about sprouting. Or indeed, it could just be about synaptic plasticity, and it could just be about what we do to the remaining neurons. Now, one of the difficulties I think we had, and my friend Lisa, the problem with Martin is that conceptually they said what no-go does and what the iron-1 antibody does is it enables an axon to bridge the scar tissue and then grow through new undamaged tissue and find a functional connection. And it became clear that that just wasn't the case. That wasn't really happening. A handful of neurons seemingly were doing that, but usually what they were really looking at is they were looking at spared neurons, so incomplete transections. And it was a very small, so nothing, that wasn't really happening. And the problem is that Martin, right at the outset, said that he wasn't interested in this. He didn't think this was the way it was working. Now I think everybody admits that actually it's not about regrowing, it's about sprouting. So that's just something to bear in mind. So in terms of if you want to regenerate the spinal cord, you're probably looking to encourage this and this rather than this. Okay, and that really does change the view of what's actually going to be necessary and what stem cells, for example, could be doing, or any therapy could be doing when you're trying to get some sort of functional recovery. Complete transections are always problematic. Incomplete, where the ability to sort of improve function is something that I think that there's an enormous amount of these sort of interventions. But it still remains problematic because clearly, if we can imagine regrowth, finding its target, let's say it's a lower motor neuron and we've damaged a motor neuron. And you can imagine, okay, it has to find its target and start activating it. But plasticity, again, slightly problematic. And this is actually probably where some of the difficulties arose with those NGF injections, is that actually it generated unwanted synaptic connections. And then these gave rise to those sort of pain syndromes. So yes, let's regenerate. If we don't even start to trigger this phenomenon, we will never repair the spinal cord. But I would just like to say right at the outset that it's only the beginning of what you have to do. You then have to worry about how you get the appropriate sprouting, the appropriate synaptic connections. Now, that's not to say that elsewhere in the brain there hasn't been quite a lot of success, and with stem cells. Many years ago, we worked with some people in Lund, in Sweden, Anders Björklund and Oli Paulson, no, I've got it slightly wrong. And these guys took embryonic stem cells and they injected them into these are sort of fetal tissue that were dopaminergic neurons effectively that were put into induced Parkinson's disease. And they did something really interesting. They did seemingly produce new functional connections, but the problem with the striatum and dopaminergic neurotransmission is it's very modulatory. So that's principally how we think that was so successful. But it does, and there was a reason why I wanted to mention that, which is kind of interesting, is that was basically tissue that was transferred from one individual to another individual. So basically it's non-self. Now, the interesting thing, and one of the fascinating things about the central nervous system that was recognized about 1946 by a guy called Peter Medwer, and he was able to show that you could transplant tissue from one organism to another and not have rejection. And this is something that's really quite interesting about the brain, is that if you do inject cells directly into the spinal cord, it's not so much of a problem. Although the cells I'm going to talk to you about, well, then they weren't autologous, clearly, because I'm going to talk about cells that went from a human to a mouse. But we weren't concerned about that. First of all, the time frame is short, but likewise, injecting xenotransplants from a human to a mouse actually doesn't seem to matter that much, just because of the way that the immune system works in the brain. It's very, very different. So we've got an environment which is basically very inhibitory. And it wasn't just about no-go interacting with a no-go receptor. It now transpires that the brain really does, and the spinal cord goes to a lot of lengths to try and prevent that sort of regeneration and sprouting. So MAG, for example, a whole series of other cell surface proteins that basically trigger inhibitory processes and cause collapse of these growth cones. Let's just have a quick reminder also about the nature of the damage, and this also has ramifications, and it is important to thinking about what actually these stem cells could be doing. So if we imagine that the stem cell isn't there necessarily to generate new functional connections per se, but it could be doing something to try and prevent some of the sequelae associated with spinal cord injury. So if you have a spinal cord injury or traumatic brain injury, fundamentally, that injury site, you're going to kill neurons, they're dead. But one of the things that I've studied for many years is that actually, if we start looking a little bit outside that region, we suddenly discover that there are all sorts of things that happen downstream in the central nervous system, where we end up with secondary damage to neurons. And that secondary damage, as we've heard throughout the talks this afternoon, is that actually, if mesenchymal stem cells, or whatever stem cells, or a mixture of hematopoietic and mesenchymal, if they're doing anything, it might be actually to modify the way that the immune system is behaving. Possibly even the acquired, we heard today, which was interesting. We tended to always feel that this sort of, the role of the stem cell might be to modulate the innate immune system. So neutrophil function, macrophage function, the idea that it could sort of influence the way a T cell or a B cell is behaving is something that's a little bit left field, actually. So it's kind of interesting hearing that today. But fundamentally, if the stem cells, by doing whatever they do, blebbing, generating exosomes, extracellular vesicles, or by somehow conditioning the host, it might reduce the susceptibility or the amount of proteases that are present in the inflammatory lesion, free radicals, cytokines perhaps, all of these things. A neuron is particularly at risk for all of these components. A neuron has to try and stay alive. It needs to have trophic factors that come from above and trophic factors that come from below. And if we transect that axon and there are no collaterals, that neuron will die. So if we can try and prevent the susceptibility of those axons in the spinal cord to this damage, you might get survival of the neuron, and you might be able to suppress the inflammatory response with these stem cells. The other factor that's happening, and maybe there could be some modulation, is, and we talked about energy metabolism as being really important as well, and promoting energy metabolism. So one of the ways that neurons, neuronal cell bodies are also going to die, that is distinct from other forms of cell death, is excitotoxicity. Excitotoxicity is a strange phenomenon. I mean, it goes back years and years and years. Some of the first experiments we did trying to investigate the unique features of inflammation of the brain involved injecting canic acid or NMDA into the brain. These are glutamatergic agonists, and because they're not properly cleared by the brain, like glutamate, what happens is that it can execute a neuron. So that's excitotoxicity, and we can imagine that we basically get an influx of calcium into the postsynaptic nerve terminal, and if we do that too much, we activate neuronal cell death, which seems a bit counterintuitive. Now, oddly, what will happen in an ischemic lesion around an injury site is that you get to the point where the astrocyte, which normally clears the glutamate from a synapse by pumping it into the astrocyte, converting it to glutamine, giving it back to the neuron and allowing it to be released again. What happens in a scenario where there's not enough ATP is that pump reverses and it starts pumping glutamate back into the synapse. This is a really crazy thing to do, and basically the brain executes itself once again. So again, if you can supply substrate, or you can meet the energy demands locally or encourage to reduce energy utilization locally, this also might be a way in which you can manage to salvage tissue. This, of course, and these features tend to be thought of being very acute, and they'll happen. And the sad thing for things like traumatic brain injury, for stroke, for spinal cord injury, everybody thought, well, if we just block these receptors, we stop the calcium influx, we're going to salvage the spinal cord and the brain. And that has never worked. Partly because you can't get in there early enough with these blockers to actually make a difference. On the other hand, we know that there are metabotropic glutamate receptors, and this is particularly important in the context of Alzheimer's disease, where actually a low-grade, low level of excessive glutamate production and a downstream response that is modulated by these mGluRs might actually play a role in some sort of longer-term excitotoxicity. So again, it may be that actually this is ongoing perhaps, but in terms of whether this would be good after a spinal cord injury or brain injury, the principal problem is that it misses the window. So the excitotoxic response, the most of the excitotoxic response is about here. And then downstream, we get recruitment, activation of microglial cells, recruitment of neutrophils, and recruitment of monocytes, all part of an inflammatory response following the spinal cord injury. And that might be something that we can manipulate, and maybe stem cells will have an impact on that. I should say right now that methylprednisolone has been given to spinal cord injury for many years. Every single study that has shown, all the sort of the larger studies have shown that actually it doesn't work. So that immediately makes you say, well, why would you focus on this when you know that methylprednisolone doesn't work in terms of an anti-inflammatory strategy? If steroids don't work, nothing will work, surely. But then, of course, the difficulty is that steroids have rather profound effects throughout the rest of the body, and what you want is targeted delivery and something happening in the spinal cord. So we'll just hold that thought for a moment, but I mean, it doesn't mean perhaps inflammation isn't the actual be-all and end-all. Anyway, this is just highlighting how it is that after a spinal cord injury, so this is one of our animals, and we've damaged the spinal cord with a contusion injury, and we see that 24 hours afterwards, we get a lot of neutrophils recruited, and it takes a very long time for them to get into the tissue. But that's useful because it gives you a window where we can try and inhibit that influx of neutrophils. There's more debate as to whether or not a monocyte is a good or a bad thing in your spinal cord. So we're going to stick with neutrophils for the moment. And let's just have a look. This is this is a molecule that is chemoattractant for neutrophils. And what we have shown effectively is if we inhibit neutrophils from being able to enter the spinal cord, we get a massive decrease in the amount of blood-brain barrier breakdown. So this is injecting CXCL1 into the spinal cord here and here, and if we remove all the neutrophils from the animal, we discover that all that blood-brain barrier breakdown goes away. So neutrophils basically are bad news in the spinal cord. Right, so I'm presenting a background to you all for thinking about how stem cells might work if they're not actually integrating and making functional connections. And I'm showing here that actually the inflammatory response is bad news, and certainly in the context of a neutrophil. So now let's have a little bit of think. And this is sort of recapping, Boris focused on this, and I just think it's worthwhile highlighting again problems, and I'll tell you again a little bit about some of my experience in the past. We always say stem cells are great and important, and we're hearing how and most of the people here, of course, have a vested interest in stem cells and think they're probably a good idea, but it's worthwhile just pointing out that it's been 20 years since OECs were injected into the human cord, and we still haven't seen any positive outcome. It's more like 40 years since Bill Blakemore was injecting Schwann cells into the spinal cord and showing that he could get some regeneration if he did that, and nothing has translated. So I'm afraid, but this was at a time when everybody thought the whole point of the stem cell was to encourage regeneration, but also to enable that axon to cross. So anyway, now I hate to say it, is that we've had approximately 40 phase 2-3 clinical trials from mesenchymal stem cells, and very few go on to phase 3. So as much as we're seeing lots of good things, we have to think about how we're actually going to progress. One of the difficulties, obviously, and this has been highlighted again, and I'm going to talk to you about the neurocells and why we think they're a good idea, because basically they're naive cells principally. You said virgin. I'm not entirely sure that's a good word to use to describe them. It's cute, but I wouldn't go there if I were you. We need to think about what sort of stem cell we're going to use. Should they be cultured? This is where we get to this idea of naive or allowing them to proliferate. And then where are you going to put them? Right, we've already heard the arguments for this. Should they be cultured and what type of stem cell? Let's think about how they should be delivered. If you inject anything intravenously, it's going to end up as cell. It's probably going to end up in the lung. And if it makes it through the lung, it's probably going to end up in the liver. And then the idea that it's going to find your injury site is pretty slim. Now, it's quite one of the reasons I talked about in one of the questions. I said we were trying to target extracellular vesicles by putting markers on the outside of them, so they'd go to the brain, to the spinal cord. But fundamentally, as I mentioned yesterday and again today, the brain is one of these places where it really doesn't like to encourage the recruitment of new cells, especially not immune cells, especially not hematopoietic stem cells, and probably not mesenchymal stem cells too. The last thing you want to do is have a whole massive influx of cells that you didn't want into your brain. The brain has always been really self-contained. So again, it may be that we're always going to have to inject the cells into the brain. If you put them into the CSF, I promise you that's not a route to get them to your target either, because the CSF flows from the lateral ventricles all the way back out and over the top, and who knows where the cells go. In fact, we have injected them into the cisterna magna before and not been able to find them in the brain parenchyma. So again, sort of highlighting some of the issues. So how are you going to actually get the cells? In other peripheral organs, possibly less of a problem, but the brain is pretty much designed to repel stem cells. And so then, of course, one of the difficulties is we have to think about how many stem cells as well. I haven't read how many, but again, it's very unclear what I've said. You've got 100 billion neurons in your brain, and sometimes we talk about putting a few million into the spinal cord of the brain, and suddenly you realize that this is a ridiculously tiny amount compared to what you're trying to repair. You take out a chunk of the spinal cord, and I promise you, there are more cells in that little chunk than you can count. You certainly considerably more than 1 million, let's say. So then there were these cells that we've all given up on pretty much, I would say. I mean, if you look around the literature now, these are the stem cells that I started off playing with when I was working with Martin and Alicia Schnell. And we took these green fluorescent protein positive embryonic stem cells, and we put them into the spinal cord of rats, and we had some interesting results. They seemed to sort of do all sorts of things that made them look a little bit like neurons for a little while. And then the animals developed these nasty tumors, nasty, really big, visible green tumors in their spinal cords. And everybody said, "Oh, well, that doesn't matter too much. The point is, we got one neuron to make a functional connection." And then you say, no, that's actually not useful. The problem is a tumor. There might be ways of preventing that from happening. And it may be that iPSCs, for example, might present that more utility. The difficulty is that you de-differentiate, then you differentiate. Once you've differentiated, the cell doesn't want to move around anymore. If you pre-differentiate a cell and make it a neuron, it's not going to move around. If you pre-differentiate it into an astrocyte, I don't know whether that's this is problematic. If the cell is basically undifferentiated, then it has the potential to do all these sorts of nasty things and form teratomas and anything else for that matter. So then you come, and the reason why so many people use mesenchymal stem cells and hematopoietic stem cells is because fundamentally, although they have the ability to differentiate in many different cell types, they've not been shown to form nasty tumors on the whole. But we do see that they seem to have sort of very different characteristics. So mesenchymal stem cells can basically transdifferentiate into ectodermal and mesodermal images, which is kind of interesting. Again, neurons are highlighted, but that actually doesn't seem to be the point of what we're trying to do. Right, so these are sort of the relative merits. this does seem to be safer than many of the other approaches that are being adopted. So let's just have a quick look at how they've been used. So this is some neurocells, and this is a sort of a typical, and this is not my data, I'm not responsible for it, this is Hans' data, and Tatyana will be on the next slides. And I'm just going to show you how they've used them to a great advantage. Again, it's an animal model, and it's a bit of a surprise, but these are experiments that were similar experiments to the ones that were meant to be happening here today. And what we're looking at is an interesting animal. It's the FUS mutant animal. So sometimes described as a sort of a model of motor neuron disease. It's a very small number of individuals with familial motor neuron disease and that are have this particular problem. And we can see that following the injection of the neurocells into these animals, we saw positive effects both in terms of the appearance of the muscle. So when neurons die, the muscle begins to waste. Sometimes it's not clear whether it's the muscle that goes first before the lower motor neuron. But in actual fact, the outcomes with the neurocells look very good. And we see that the percentage of mice that are able to succeed in the wire test is dramatically well, we've got wild type here, and then we've got neurocell treated FUS mutants and the FUS mutants. So we're comparing this with this, and we see that actually the neurocells have had a positive impact on the outcome of this quite profound neurodegenerative disease model. These animals die, and it doesn't stop the animals dying, but they do. They certainly do better. It's also been shown in another model of motor neuron disease in the SOD1 mutant, which is probably one that you'll all be very familiar with, I expect. Where once again, if we look at the wire test, and we see that in the neurocell treated animals, percentage of mice that fall is diminished. We see improvements in muscle weight, food intake, and liquid intake. So the SOD1. So for ALS, it looks like these neurocells are having a positive effect. Right. I'm not going to, don't know why that's there, because that's the slide you've already seen. So let's now have a little look at traumatic injury. And this is a slide using neurocells on the BBB score. This isn't blood-brain barrier breakdown. This is a scoring scheme for the ability of mice and rats to walk after a spinal cord injury. And this is the sort of work that drove everybody to think that this would probably be a good idea to use in human spinal cord injury. And I should just point out that the difference between the blue line and the red line doesn't look very great. But in terms of what that means in terms of the BBB score, these animals are certainly more functional. It's the ability to actually support yourself a little bit while you're getting about, as opposed to not being able to move around. So it's a non-linear scale. That's the problem with the BBB scale. So there are differences here and here showing that the cells were effective. And other indices in terms of what happens with the production of IL-1 beta cytokines, for example, we see that there may be an inflammatory component that's being suppressed by the neurocells in terms of how they might work. Right, I'm just going to scamper then through the new unpublished data that we've just got, highlighting how these cells that we're going to go into the Alzheimer's animals worked in a TBI model. So what we did, it's very acute. We took that spinal cord injury model I showed you right at the outset. We just, instead of hitting the spinal cord, we take a chunk off the top of the animal's skull. We smack the animal with a large area. It's a 2 millimeter diameter rod that basically displaces a bit of the cortex. And then we glue the bone back on. So in actual fact, what happens, one of the best therapies for a spinal cord injury is just trepanning. So basically, the spinal cord injury, brain injury, is to remove a bit of the skull. So we put the skull back on after we've injured it to make sure that all that edema can't dissipate and causes all the damage that needs to damage. So it's much more akin to a human TBI. And then we decided to kill the animals basically at 48 hours after the injury, and we've injected the base of the lesion either with some saline or with the mixture of HSCs and MSCs from neurocells. So harvested from an individual, flown to Oxford, trained to Oxford, in fact, and then we very rapidly, the whole thing has to be well coordinated because the cells only last for a certain amount of time. And then we injected them, did the lesion injected them. We injected them either at T0 or 6 hours after the injury, and then killed at 48 hours with some behavioral tests in between. And what sort of results we get? 24 hours afterwards, the animals were a bit upset about having had the TBI. And what they all do, if we look at the neurocells going in, so that's T0 or T6 against the control TBI, and we see that actually there are seemingly no differences. The animal, you can tell the animal was hit on the left-hand side, left hemisphere injury, and therefore the time for them to take some sticky tape off their paws was reduced in the animals that were on their right paw, the contralateral side. It all makes sense. They were able to get it off on the left paw. They noticed that they had tape on their paws at exactly the same time. So they knew there was a problem, but they just couldn't get their right paw tape off. That's not significant, but actually what we did notice is if we look at the controls and how many, they're given 120 seconds to take off the tape. And if we just say who was successful and who wasn't successful, we realized that on a chi-squared, actually, the right paw animals that had received neurocells basically did better because more of them actually managed to get the tape off. So that was, we didn't know this until after we did all the analysis was done blind by 3 individuals who had no idea what was going on. They just measured the time it took the animals to take off the sticky tape. Here we also had a marble burying test, an anxiety test where the animals scamper around and bury marbles. But here, this is where it really looks really very, very striking. And we look at, here's the control, here's the right paw again. With the left paw too, it seems that we're getting some sort of strange improvement as well. Not a bit unexpected here, it's not significant. But if we look here and we're comparing this time the T6, but we can see that, I don't know, we're missing significance. Unless you do, this is a two-way analysis of variance. Interestingly, if you cheat and you just compare this with this, it's significant. But we can see here at 6 hours, we definitely get a very, very significant effect of the neurocells on function. And here we're looking left paw and right paw, and you can see how if we decide that we do them independently, then obviously it looks better. But this is the proper stats, and that's a really robust result. In terms of what we could see in terms of the lesion at 48 hours, it was somewhat less convincing. The lesion weights of the animals were unchanged. The lesion area doesn't look like it was much different. I mean, and so it's hard to equate exactly how that loss of tissue then relates to the functional effects. But what we did see is certainly in this particular instance, there was a reduction. If they were given a T0, there seemed to be a reduction in blood-brain barrier breakdown. That's significant. That dissociation, though, because obviously we saw the 6-hour time point, means that maybe the blood-brain barrier breakdown and the degree of edema actually isn't as important as we thought it was, because we certainly saw good functional outcomes in the animals treated at T6. So fundamentally, and to a skeptic, we've had to. It's been shown that in spinal cord injury, these cells are effective and have a significant effect on outcome. Now turning our attention to traumatic brain injury, for which there is again just no therapy that seems to work, no effective therapy that you can give. We find that actually the injection of these cells seems to have a really quite a useful impact. I shouldn't use the word impact, has a useful outcome at 48 hours. And while the area of cell death wasn't affected, maybe there was some advantage in terms of blood-brain barrier breakdown. So that still leaves us with the old question. We all sit here and we don't know how it works. So the lesion area wasn't affected. We need to go back in here and we'll have a look at the inflammatory response, etc., etc. But I think that fundamentally we're still in this community, we're left with this problem that we like to play with stem cells, but we know that they will die and disappear fairly quickly or we can't find them again. And yet they've left behind some sort of echo well, they are the echo and of some really sort of rather interesting positive outcomes. So I'm going to leave it there. And I think all of us have to think about how we can find out where our cells went, find out which bit of the cell it was. And one of the interesting things I was going to show you before I decided not to talk about the microbiome today is I'm going to show you some work we've done where we were giving basically probiotics to animals, and we discovered that it was actually the dead probiotic worked just as well as the live probiotic. And I was chatting yesterday about how some friends of ours in Argentina showed that basically dead stem cells work just as well as live stem cells. So I think this is one of those questions, is it about the host response or what the stem cell actually does on its own? And I think that the more we learn, the more we realize it's probably more to do with what the host response to the presence of the stem cell. Great, leave it there. Thank you very much, everybody, for putting up with me.
Thank you so much for this brilliant talk.
Any questions?
Some questions, just comment. There's one of the questions there, do that cells go, do they go to the site of injury or they go, they're aware go to lung? So basically, different set of experiments. We made created stroke, brain stroke by occlusion of the brain activity.
Yeah.
And we inject cells which were translated with GP.
Can you put them IV?
Yes, we put them.
Tell me.
Yeah. And basically what we saw, really, the substitute cells go to lung. And it's healthy brain, they never go to brain. But now the rats with lesions, they really go to brain. We saw it next day. So there, we still have a lot of cells in the lung, in the liver, but we did saw cells in the brain. We saw definitely the next day. We saw it in 3 days, I think. And they disappeared after 5 days for sure.
Okay,
But we did that
so those are nice experiments to do, and I think that those are essential things to do. It is always disappointing, but if you look at the relative percentage of the cells, how few cells end up in your target tissue though. So if they don't end up, what we also find is they don't end up in the lung, they'll end up in the liver. So if you look at all the organs, you'll find that you can try and recover all the cells and find out what percentage you actually the percentage of the injected dose. And then it's tiny. So exactly. So I'm coming back with sort of yesterday I talked about briefly about how with the metabolome, one of the work that I think best known for in Oxford is showing that after you enter the brain, you make lots of changes in peripheral organs. And sometimes if you manipulate the peripheral organ, so for example, in our case, if you remove Kupffer cells from the liver, you basically reduce damage in the spinal cord and you reduce damage in the brain. Because the liver can't respond, it doesn't generate a normal APR. I also wonder whether in these scenarios, whether actually, if you are giving a systemic injection of stem cells, whether or not it's actually suppressing that the fact that they end up in lung is changing the way or redirecting an immunological response, or maybe you've got a distraction. So you have an injury in one place, you have a stroke, and instead of having the damaging consequences of inflammation, you have neutrophil recruitment, for example, to the stroked out bit of brain. Now you can say, oh, there's something really weird going on in the lung. And so it could be that we're sort of just redirecting immunological responses in some strange way. These sort of questions haven't really been properly answered to understand how it is. And clearly, of course, some of the cells that you saw making their way to the territory of the middle cerebral artery, they might be doing something useful there, although we don't know what they're doing still. So useful, yeah.
They never go to healthy brain.
No, well, that's right. But I think that's all to do with the other interesting. Just some friends of ours have also done some stem cell research after that. Do you know how they found? They found the cells in the venules. Now, it might be interesting to have a look back and see where your cells are. So you have to imagine you just tied off the middle cerebral artery, and you can find some cells in the venules. So how did they get there? They must have gone backwards up the circulation. Which is fascinating. They can't come through there's no arterial supply, and yet they were finding that. So again, it might be fun to go back and have a look at that tissue and decide where they were.
We didn't cut the artery. We just put there.
Oh, you did the wire. So you did the transient. Transient ones.
Stroke and the dreams.
Okay. Well, in the permanent, people have seen that phenomenon in the permanent model where. So basically ligation of the middle cerebral artery, and they've seen stem cells in the venous in the venous supply, which is a bit of a strange thing. But you're right. If you take away the inclusion, then they can have access that way.
All right. Thank you so much. We have to move on. And I think we still have a chance to discuss a little bit during the meeting, maybe more after the session.
Our next speaker, Professor Evgeny Kureysh, from Biology Department, School of Science and Humanities, Neurodegenerative University, Kazakhstan. Please,
Maybe, I don't know. I think I'm loud enough without microphone. I don't know how to.
Right, maybe it's.
Okay,
More loud than women, yeah,
yeah.
in general.
So how to? How to switch it? Probably just I slowly start. So first of all, thank you organizers for giving me opportunity to present my talk here. And actually, I'm at Nazarbayev University for less than a year. I'm a new person. It's really nice to basically talk to or know people better from National Laboratory of Astana. Because at Nazarbayev University, we do a lot of teaching. And because of my tight teaching schedule, actually, organizers agreed to give me an opportunity to present my topic today, because today I will not talk about stem cells. Actually, I will talk about platelets and complement, about some immune components that contribute to pathology of epilepsy. Although my topic is not really related to stem cells, actually, I can talk that platelets actually play a very important role to sense the injury in the CNS. And actually, it's known that the platelets then produce a lot of factors actually to attract stem cells to the site of injury, especially in the CNS. But I will probably come back to that a little bit later. So if you think about epilepsy, that it's that's it's really enhanced level of electric activity of the group of the neurons, and most of the cases you have a focal areas when you have some, and typically epilepsy happened as a result of some kind of injury. So it's a result of improper repair. Although in some cases, like 30% of the cases, you do have a generalized seizures. And the mechanisms, while actually it happens, that's our current knowledge suggests that we do have an imbalance between excitatory neurotransmitters like glutamate and inhibitory neurotransmitters like a GABA. And the reason why actually we know about it, that's because it was a drug. Actually, it's PTZ or actually metrazole. It's actually GABA receptor antagonist. It's quite interesting. This drug was designed to treat epilepsy because in 40s, many scientists believe that epilepsy actually, not epilepsy, not treat epilepsy, it's to treat depression. And actually, because in the beginning, many scientists believe that depression happened because we have change in balance towards actually more inhibitory symptoms, more GABA, and less actually glutamate. So they try to treat depression using this drug, and then they found that it can cause actually very severe convulsions with very severe seizures. And indeed, actually, this drug now FDA revoke license for this drug. So I think it's one country in Europe, I think Netherlands, this drug is still can be used actually for some of the treatment. But anyway, it was revoked, and now it's used actually. It's a very good model to induce epilepsy, especially in different animals, from primates to rodents to the mice. And here just a concept that you do have a inhibitory synapses, and they kind of compete with excitatory synapses. We have both these systems. But what is quite interesting, when you have any type of the injury or some kind of severe neurodegenerative disease like Alzheimer's disease, for some reason, every time with traumatic brain injury, after that, when you have a repair of the CNS, actually GABAergic neurons actually more prone to the death, and usually they repair in much more slower rate. So after injury, often you have enhanced electric activity. After injuries like in Alzheimer's disease in very severe stages, you do have actually a lot of epileptic seizures. So now we have actually tons of anti-epileptic drugs which can actually change the balance by just blocking excitatory receptors or actually by enhancing inhibitory receptors. But the problem with all this therapy, that's about 30% of the patients with epilepsy that don't respond to this type of anti-epileptic drug. So which means we have to understand better what's the mechanism, and is it just a disbalance of neurotransmitters? Maybe there is something else. What happens when you have epileptic seizures? Actually, now what actually shown here probably have seen these pictures many times, but I just made it by myself. This is the synapse. This is the presynaptic terminals, and this is the postsynaptic terminals. What is interesting, and actually you know about it, you saw these pictures many times, and here we have vesicles, we have release of neurotransmitters, and blah, 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 and here we have receptors for neurotransmitters. But probably what you didn't think about it, that postsynaptic membrane is quite interesting place. This is actually very stabilized, very thick membrane, which has actually a lot of cholesterol, and it has a lot of glycolipids. About 8% of this postsynaptic membrane actually consists of not of glycosylated proteins, as many people think. They consist of multiple glycosylated lipids. And majority of the lipids, they're unique to the CNS, because other organs, they have different lipids, they have different sugars on the surface. And this is actually a quite interesting picture. You have neuroblastoma cell, this cell is not differentiated, and this cell start to differentiate. And you can see how lipid troughs appear. There are specific toxins, or many actually bacteria, they make a toxins that specifically bind to these lipid troughs and modulate their function. What actually we found a while ago, that platelets can recognize this brain-specific lipid troughs. Actually, you can even isolate brain lipid troughs from the CNS and inject them intravenously, and mice die because of anaphylactic shock. And the reason for that, because platelets can recognize lipid troughs with specific receptors, and first they degranulate, they secrete a lot of factors, actually including factors which can attract stem cells or immune cells. And finally, they even blown up in a very large number of microparticles. Again, these microparticles, they have a lot of repair function, they have function to basically attract immune cells and do many other things. And what we found that a lot of these lipid troughs are, of course, in postsynaptic membranes, on the receptors, on neuronal endings, and also on astrocytes. You know, the blood-brain barrier has two lines of defense. One line of defense, you have tight junctions of endothelial cells, and second line, you have tight ring of limitants of the astrocytes. And actually, a lot of lipid troughs here. So as soon as you have any damage of the blood vessels, platelets get outside, they can recognize the lipid troughs, and they produce a large amount of different factors as shown here. So that's actually the mechanism how immune system of our body sense any neurovascular damage in the CNS. And I just want to remind you that CNS is highly vascularized and have very high density of the vessels. Quite interesting, I already told you that lipid troughs can be isolated from the CNS, which is quite simple protocol. You just homogenate whole brain in mild detergent. And then you have actually pieces of postsynaptic membranes. And then you can basically put platelets and these pieces of membrane together. And that's quite interesting that it's known that platelets can secrete a lot of neurotransmitters. And we actually tried to measure all of them. And we found that the only neurotransmitter which is secreted by platelets, if you give them neuronal lipid troughs, it's serotonin. They really produce a large amount of serotonin. And of course, we thought maybe it plays some role. For example, in epilepsy, where the balance of neurotransmitters actually is known. Although actually serotonin is not really well known. Is it protective or it doesn't contribute to epilepsy? Because from the one hand, people who take antidepressant, they're less prone to epilepsy. On the other hand, if you suddenly increase the level of serotonin in your body and your brain, you have so-called serotonin syndrome. When you have a tremor, you have also enhanced electric activity. So it was a question. So we decided to use this is a specific setup. You can put electrode on a mouse head, which can basically sense electric activity of the neurons. And actually, mice are freely moved in a cage. There is a specific receiver here, that's Faraday cage. And then you can see neuronal electric activity. So the first experiment that we did, we just depleted platelets and induced epileptic seizures. And the result actually was quite shocking to us because, of course, in control, when you check with PTZ, you get nice seizures and convulsions. And when you treated platelets, it was basically nothing. If you look at the number of high spike peaks, they were dramatically reduced. So mice actually feel actually quite good. Another interesting fact that we found that indeed, if you don't deplete platelets, you do have a spike of serotonin in the brain after the seizures. Within 10 minutes, you have quite substantial increase in serotonin concentration in the brain. But if you deplete platelets, it doesn't happen. And another interesting fact that after the seizures, blood-brain barrier become open. You can look at by specific dye. And if you deplete platelets, amount of opening BBB actually is decreased. It's very similar to that Anthony told about neutrophils. It looks like also platelets, they kind of contribute to opening of BBB, of blood-brain barrier. And here, I don't know, can you see it or not in this monitor? We just did staining for blood vessel, for neuronal blood vessel with a lectin, and also staining for the lipid rafts, and we did find actually some of the platelets outside of the blood vessels close to neuronal lipid rafts in the sense, platelets are supposed to be red here, lipid rafts here are supposed to be green, and the blood vessels also here is stained in green, and you can find platelets outside. I don't know. Well, if you cannot see that was published, you can look at the paper. Yeah, so I guess at that point you have to believe that indeed you can detect the platelets outside of the blood vessels close to the lipid raft. And finally, it was quite an interesting experiment. We decided to inject platelets straight into the brain. Again, Anthony, we got a lot of talks about it, that brain typically doesn't want to take a lot of things, it expel everything. But if you inject it directly, again, the result was quite shocking, and the mice developed very severe seizures. If you then, because we did experiment when I tried to inject just the plasma, it didn't happen. You do need to inject intact platelets into the CNS at the site of injury, and you do have actually very high level of electric activity, and you do have some spike of serotonin after injection of platelets into the brain. To make long story short, we did actually a lot of very sophisticated experiments for adaptive transfer of the platelets with depleted serotonin. Actually, we found that indeed serotonin was a key neurotransmitter that's enhanced electric activity of the neurons. So that's kind of new model. And maybe eventually right now we'll try to make some kind of trials and do some experiments. We'll try to treat epileptic seizures with aspirin, so some kind of anti-epileptic drugs. So actually, in animal models, we should quite work. It works, but we'll see how it's gonna work in humans. So here it's a model that's when you have some kind of CNS insult, like epileptic seizures. BBB can become compromised, and then platelets enter the CNS, and they interact with the lipid raft, and then produce serotonin and other factors, and that basically propagate the pathology. And the next question was, since we know that the platelets enter the CNS, I've been interested in this immunologist, what kind of cells can react to the damage quite fast. And actually we have platelets that react within seconds in the damage of the blood vessels. And there is another system which we have, which can actually react to the presence of some pathogens in the blood or have some damage, is a complement. So when you have opening of BBB, complement can enter CNS. And actually complement can also be produced in the CNS. Some components of complement can be produced by astrocytes, and you know that classically complement can bind to specific sugars on the surface of bacteria, or it can bind to antigen antibody. So if you have some kind of antigen on the surface of pathogen, you have antibodies, it can bind. This is C1q. In fact, actually, C1q can bind to the surface of the neurons, and in this case, it's less known what they particularly bind. For example, there are several candidates like pentraxin or phosphatidylserine. The interesting fact that indeed, if in the periphery you have antibody-antigen complex, and then activation of the complement, C1, then you have C4 activation, C3, and finally that would lead to formation of membrane attack complex, specific holes on the pathogen, and also it helps to opsonize or cover pathogen to enhance phagocytosis. What happens actually in the CNS is also quite interesting. We do have activation of classic pathway, C1q, which is here, can bind to the surface of the damaged neurons, and then C4 get activated, and then C3. And then it's called to interesting process called opsonization of the synapses. I remember I showed you this synaptic membrane. It was shown quite recently that indeed during the development, if you have non-electrically active or extra synapses during the development, they become covered, opsonized with the complement, with the C1, C4, and C3, and these non-functional synapses get eliminated by activated microglia, which have complement receptors. And that's during the normal development what happened. But what happened to during certain pathology like schizophrenia this elimination is actually too robust. Too many synapses get eliminated. But actually, autism is the opposite. Very few synapses get eliminated. So that means that indeed C4 might play a big role in pathologies like autism and schizophrenia. And this is genetic analysis for schizophrenia. You can see that C4 actually is quite the chances that it involved in this pathology association with C4 is actually quite high. You have a tower when compared to other risks of the gene. Of course, the question is how it works, how complement or C4 works in case of schizophrenia or autism. If you look at the genes, C4, human has actually two variants, C4A and C4B. They're quite highly polymorphic because they're located on MHC+3 locus. Highly polymorphic. Also, they have actually specific retro genes insertions which can become activated during inflammation. For mice, it's a little bit easier. They do also have two genes, but only one gene is active in complement cascade. So it's much easier to look at the mice and see what actually happened during epilepsy, whether C4 or C3 play a role in that. So here again, I just say that if you have electrically active synapse, these synapses express a lot of proteins which can destroy or remove complement. But if it doesn't happen, a lot of complement is produced by astrocytes or got from the periphery during injury, and they cover these non-functional synapses, including presynaptic and postsynaptic membrane. And especially been interested what C4, if it's so important for Alzheimer or autism, play a role in activation of the neurons via this postsynaptic membrane. Design of experiment was quite simple. We took wild-type mice, C3 knockout, and C4 knockout. I already told you the mice only have one gene, C4B. And that's quite interesting that C3 knockout mice, they have even less severe. This is epileptic score, 6 is a death. Here we have milder symptoms. So anyway, C3 knockout mice, they even have a milder manifestation disease. And this data is still unpublished, very, very, very good data. Whereas if you look at inject standard dose of PCT into C4 knockout mice, they'll die within 10 minutes. And it's even more dramatic if you use subconvalescent, which is low dose of PTZ. If you inject this amount to wild-type and C3 knockout mice, they don't develop any seizures. Whereas C4 knockout mice, 80% of them, they die again within from 8 to 12 minutes. That was quite dramatic. They actually didn't respect this result. First, what we tried to look at, it's called neuronal immediate early response genes. Probably the people who are in the cancer biology field, they know that there are specific receptor tyrosine kinases which can bind specific growth factors, and they can produce, they can actually activate expression of the gene within the minutes because they don't require additional protein synthesis. They already use kinases which are already there. So they go to the nucleus, and the first you get expression of these immediate early genes, and then they further start induction of late response genes. So indeed, if you have enhanced selective activity of the neurons, as shown here, so for example, glutamate receptors, enhanced neuronal electric activity should increase expression of immediate early genes within the minutes when we inject PTZ. And again, if you think about RTK, receptor tyrosine kinases in the CNS, there are two of them, which have been already mentioned, which are very important for repair of the CNS. It's TRK-A and TRK-B, which are receptors for AGF and BDNF. These are the most important factors for CNS repair. Here, what we found, indeed, if you take wild-type C4, C3, and 4 knockout mice, C4 deficient animals, they have even very low level of baseline expression of early response genes EGR1. And this gene is very important for synaptic formation and for cognitive functions like memory. And if you inject PTZ into wild type, you can see this gene is upregulated quite dramatically within 10 minutes. C3 knockout mice, they also upregulated. But as we look at C4, the level of upregulation is comparable to unmanipulated wild type mice. The same true for C4. You can see that C4 knockout mice, they couldn't upregulate it, and this
Do you think that you said the complement platelets can bind to lipid drugs from the brain, right? Did you check this difference of lipid drugs from old brain, young brain, for mice? Because lipid drugs can change with age.
Yes, that's changing.
And immune complement platelets are also losing the abilities. How it works with old brain?
Yeah, actually, it's quite interesting. If you look at developmental brain, actually, it's quite again. It's also interesting about the function of the platelets. Outside of the brain, platelets typically, if you have injury of blood vessels, they recognize collagen. And in developmental brain, you have a lot of collagen and not so many brain-specific glycolipids, not so many lipid drugs. But then as soon as actually a person become adult, or if it's central for the mice, the collagen is basically virtually absent in adult brain, but it's replaced by high amount of the lipid troughs and platelets can react. And as soon as you get aging, it looks like you get opposite. You kind of lose many of these brain-specific glycolipids, and again, amount of the collagen, which basically have, especially when you have systemic inflammation around the blood vessels in aged brain, you do have again more collagen and less lipid troughs. So that's basically a quite complex way how. And actually, many papers when they look at different changes of different glycolipids during different diseases, but definitely Alzheimer's disease during aging.
When it
You have-
goes up, then it goes
Yeah.
goes up again in the age.
So it's basically you have maximum amount of these lipid troughs when you're adult, young adult, and during age again it goes down. So unfortunately, actually it's quite interesting because we did show that platelets can stimulate neuronal electric activity. If you just put neuronal culture, put platelets, they immediately enhance neuronal electric activity. And that's actually helpful for Alzheimer's disease. Because it's known if you stimulate neuronal electric activity, actually it's kind of helpful for them to repair, maybe because of early response genes actually get activated. But in aging, it looks like, yes, the amount of the lipid trough decreased and platelets can stimulate them as much as they do it in adult age. Yeah.
So building on that last comment, but also just see through in the serotonin story. Platelets obviously are incredibly rich in serotonin. If you just inject serotonin, does it get cleared so quickly that it doesn't have the same effect, or is it basically a slow release serotonin bomb? How does it? So did you just do the serotonin injection?
Yeah, I did this experiment. Yes, if you inject serotonin into brain, it can cause seizures. But the concentration should be higher than on platelets.
Yeah, that's the interesting thing. So when I said, is it like a slow release or
if you do slow release, it's not going to cause the seizures. And actually, for slow release, then you have changes in the number of receptors. And then if you're suddenly injected, then it doesn't cause seizures.
That's the next question. So what do we know receptor-wise? What receptors with? Is this like a non-specific serotonergic storm hitting everything? Or would you like to say this is a particular receptor that's responsible?
It's more like serotonergic storm affecting multiple neurons, and it's like systemic effect. So it's more like, as I said, serotonin syndrome. When people just start taking SSRIs, they do have experience, like, for example, first day, high temperature, all this tremor, all these things. It's something similar. I don't think it's affects specific receptor because the effect is quite generalized.
So I guess where I'm going with this is whether or not we know that, of course, that in the vasculature of the brain, the 5-HT1A receptor is really important in controlling blood flow, and whether or not you're actually doing something that's causing some sort of strange vasospasm locally, and this is giving rise to a seizure, or whether it's actually the neuron per se that's responding. I'm sure. I think that could be worked out. The other, in terms of those the other experiments you've done, I did wonder. I worked with kind of carpine model many years ago, and that's associated with a huge amount of blood-brain barrier breakdown, activation of the. So I just wondered whether you'd be able to make, if you did the serotonin, if you did the platelet depletion experiment in the pilocarpine model, when you just show that the pilocarpine model is basically a platelet-dependent epilepsy model, it would be kind of an interesting. I might predict that it was just because of the nature of the damage that I've seen in that pilocarpine model.
So what actually experiments which we did, you can try to make, for example, traumatic brain injury, focal one, but a needle.
Yeah.
And after that, actually, after, for example, 1 week, if you try to inject PTZ, you can only inject actually subconvulsant, very, very low dose. And you can induce the seizures. Whereas, of course, in unmanipulated mice, you can do it. But if you make traumatic brain injury and deplete platelet, or use even anti-platelet drug like aspirin, then you cannot induce seizures after that. So indeed, like after traumatic brain injury, you do have some activation of platelets, so you kind of get handling or some changes. And
use aspirin. I think that's an issues with epilepsy itself. But for clopidogrel, it might be an interesting experiment to use clopidogrel.
Yeah, we try. Actually, that's the point. We try to use different anti-platelet agents. Yeah, exactly. Aspirin actually is more. How to say? You have more data about aspirin. Actually, even anecdotal studies about. And actually, there are some anecdotal studies that show that indeed For example, some person took aspirin for a very long time and she didn't have seizures. But then she stopped taking aspirin because she needed to get some surgery, and immediately she started having seizures. It was again quite interesting. But there are some other anecdotal, evidence.
Finish off your study.
Yeah.
I agree. And then have you injected C4 directly into the brain?
No, this is very it's again, that's would be interesting to do. Yeah.
Well, this is excellent talk. Maybe I have a very brief question of the time of C4 and C3. Are there any animal studies actually supporting the role of C3 and C4 in any syndromes related to cognitive decline?
Yes, actually, recent several studies actually in Nature and very good journal, very high-profile journals. So one study, they overexpress C4, and they found that indeed you have less synapses and do have some kind of behavior changes similar. That reminds, schizophrenia. Of course, mice don't have schizophrenia, but there are some behavior abnormal things. And they actually did a quite interesting even study. They tried to knock out C4 and then overexpress human C4A and C4B, and they found that indeed for schizophrenia, C4A is more important. But this overexpression, with the knockout, we don't really have so many studies, although there are studies in humans. They did find very strong association with the depletion of C4B gene and autism. That's that's an animal model. Again, it would be interesting to see whether these mice are kind of autistic.
In any case, seems to be it's very interesting, maybe relatively new readouts of all kinds of.
Yes, that's actually very new role. And again, all previous studies, they think that C1, C3, and C4, they play the same role. It's all synapse pruning, all synapse density. But here, kind of, we showed the first time that indeed there's something else. Especially induction of early response gene, I think it's very important because these genes are very important for many things.
Thanks again. It was really great. Right. So, if I may finalize this part of our conference. So, basically, we have done at this moment with the main parts of today. And we have a special lecture for our PhD students. So the lecture, however, will be in Russian. We have a special guest, Tatyana Khamchiev. She is a psychologist. So we're really honored to have her here. She was helping our laboratory for many years. And she will speak about how to learn in ecological way. So how our brain, the brain of students might be kind of tuned to be a little bit more ecological. So maybe as you said, it will be I have also because we have a little bit with logistics, making some of our lectures delivering soon. So I will just ask my colleagues to analyze the session.
Добрый день, уважаемые студенты-аспиранты. Меня зовут Екатерина Киктева. Я представляю сегодня компанию-организацию французскую «Тарис», которая является экологической организацией, которая занимается экологическими проектами, экологическими инициативами самых разных направлений. Что касается меня, это один из моих партнеров. Я являюсь практиком, я не являюсь исследователем, я являюсь практиком, я работаю с руководителями и сотрудниками крупнейших организаций и в России, и в СНГ, и в Европе, и в Африке, и это развитие и обучение руководителей и сотрудников по разным направлениям. У меня одна из разработанных тем – это мотивация и вовлеченность, да, работа с мотивацией персонала, это эмоциональный интеллект и развитие разных типов мышления. И меня пригласили вам рассказать о том, каким образом заботиться о себе, о своем мозге, о своей деятельности учебной, развивающий в тех условиях, в которых мы с вами теперь живем, в условиях высоких нагрузок, в условиях потока информации, в условиях постоянных изменений, да, в которых нам, несмотря на происходящее, нужно сохранять высокий уровень мобилизованности, продуктивности, результативности, да, несмотря на то, что происходит. И я буду говорить на практическом, общечеловеческом уровне. Да, моя зона экспертизы – это психология, то есть это поведение, это чувства, это эмоции, да, не те физиологические механизмы, которые под этим находятся. Итак, да, то, что совсем недавно называли вука-мире, в котором мы живем, теперь называется шива-мире. Он не только изменяющийся, разный, неопределенный, непредсказуемый, но еще и угрожающий, теперь еще и разрушающий. Да, новая аббревиатура шива-мир. И что это означает? Это означает, что неопределенность, в которой мы живем, это есть теперь наша реальность, мы должны быть все время адаптивны к этому, независимо от того, что происходит. Да, большие потоки информации будут увеличиваться, тоже они снижаться не будут, и с ними вместе увеличивается наша нагрузка, наши ответственности, наша разнонаправленность нашей деятельности, то есть это те условия, которые сейчас уже нормальной средой обитания. Да, и каким же образом себя в этом не просто спокойно чувствовать, а еще и нормально развиваться, да, и достигать высоких результатов, и бежать впереди планеты всей, у кого есть такие амбиции, да, вот здесь нужно много смотреть. Смотрите, вот если мы не учитываем то, что можно сказать экологии мозга, такой базовой психогигиеной, да, мы неизбежно впадаем в некие замкнутые круги. Что это за замкнутые круги? Если мы говорим о многозадачности, в которой мы сейчас все с вами погружены, да, многозадачность приводит обязательно к снижению работоспособности. Да, был некий миф не так давно, что можно научиться быть Юлием Цезарем и делать много разных параллельных задач одновременно. Новые подходы, новые исследования говорят о том, что нет, это скорее истощает мозг, это неэффективно, да, многозадачность приводит к падению эффективности, возрастает объем задач, которые нам нужно делать, он еще на нас нападает, да, и вот мы впадаем в это истощение. Если мы говорим о стрессе, да, в котором мы постоянно находимся в этом длительном стрессе, у нас, конечно, ухудшаются память, внимание, да, все когнитивные процессы, это приводит к еще большей нагрузке, потому что нам надо опережать, да, находясь в этом стрессовом состоянии. Таким образом, это еще один, да, стресс. И, наконец, неопределенность, с которой надо уметь справляться. Это некий современный навык, да, но, то есть когнитивно придумать, что я могу делать в ситуации, как измениться, да, вот то, что там произошло, не приехали образцы, да, нужно что-то с этим делать, с тем, чтобы ситуация не пропала. Да, если мы постоянно в этой ситуации, у нас наоборот нарушаются когнитивные процессы, да, если мы не готовы к неопределенности, да, и дальше мы еще больше, да, теряем работоспособность, ситуация неопределенности увеличивается, расширяется, да, и фактически мы теряем в принципе работоспособность, да, впадаем в то, что называется ступор. Если мы говорим, каким образом из этого выходить, конечно, могут быть тактические подходы, да, умение просто быстро справиться и восстановиться, идти дальше. Но сейчас говорят, конечно, уже больше не о тактических, а скорее стратегических, комплексных и системных подходах, то есть с тем, чтобы вырабатывать у себя компетенцию настоящего и будущего, которую называют некоторые стрессоустойчивость. То есть стрессоустойчивость – это не только просто среагировать нормально, спокойно на стресс, а уметь в длительных периодах напряжения, перегрузок, эмоций, тревоги, да, работать на высоком уровне работоспособности, да, чтобы не происходило. Кто-то называет это стрессоустойчивостью. Еще появились два таких, ну, они в психологии уже считаются и терминами, а с другой стороны, это такие некие жизненные человеческие слова. Это есть слово жизнестойкость, да, то есть это больше, чем стрессоустойчивость. И это антихрупкость. Антихрупкость означает, что из сложных, стрессовых, неопределенных и перегруженных периодов мы даже можем для себя приобретать некую выгоду. То есть это нечто, что может нас развивать, да, если мы к этому подходим системно и комплексно. Если подходить системно и комплексно, я сейчас покажу вам модель, которую можно сказать компетенциями, и по каждому из элементов можно себя мерить, на каком уровне у меня от 1 до 10 вот развитый вот этот вот навык, да, и, соответственно, смотреть то, что не развито, этому уделять больше внимания. Вот если мы смотрим, да, мы на русском назвали иронично это коктейль стрессоустойчивости или жизнестойкости, то, конечно, прежде всего это осознанность, это управляемый отклик стрессовый, да, это мы знаем, что делать, если с нами случилась стрессовая ситуация. Мы на это реагируем не автоматически, а выбирая ту реакцию, которую нам нужно. Конечно, это набор некоторых практик, которые нам позволяют быстро себя восстановить, если случилось что-то там непредвиденное и так далее. Это второй элемент. Третий элемент – это, конечно, позитивный позитивное мышление, конструктивные установки. То есть, что бы ни происходило, мы смотрим, как это на нормальную рабочую ситуацию, да, а не на что-то там экстренное, в котором мы раздавлены и не можем двигаться дальше. Это третий элемент. Пятый элемент – это вот как раз способность долгим высоким нагрузкам и неожиданностям. Это некий набор психогигиенических средств, и это выработка, да, постоянных, ну вот как тренировка, во всем нужна закалка, сноровка, тренировка, и стрессоустойчивость в этом смысле также наращивание мышц стрессоустойчивости. Если мы психогигиеной занимаемся регулярно, то рано или поздно мы вырабатываем автоматические реакции как раз устойчивости из жизнестойкости, а не из хрупкости, да, которая есть, если над этим не работать. Да, ну и, наконец, формирование экологичной среды работы и жизни вокруг нас, да, с тем, чтобы, несмотря на то, что там где-то шторм, на нашем корабле у нас комфортно, тепло, спокойно, нормально, да, и мы можем отсюда, да, питать свою энергию. Если мы говорим несколько элементов просто о базовой психогигиене, да, если мы говорим о многозадачности, то исследования показывают, что она истощает, и, конечно, резко падает работоспособность, поскольку теряется на переключение, да, если мы переключаемся на разные задачи, мы теряем огромное количество времени, из-за чего у нас возрастает объем задач, да, и мы таким образом истощаемся. Поэтому базовая гигиена – это про то, что мы отказываемся от того, чтобы быть Юлием Цезарем, особенно когда мы занимаемся учением, интеллектуальной деятельностью, то есть то, куда нам нужно погружаться. Да, директора крупных компаний, топы, которые вот еще совсем недавно была установка все время быть на связи, да, у всех мобильные телефоны, когда появились, сейчас очень многие сознательно от этого отказываются, да, поскольку это приводит к истощению. Нет, теперь у нас есть блоки времени, когда мы не находимся на связи, а занимаемся теми задачами, которые нам нужны. Да. Это что касается многозадачности. Если мы говорим, да, и, конечно, фокус на одном деле по приоритетам, конечно, да, мы уделяем, все остальное мы относим к менее значимым, да, в другое время мы это делаем. Да, если мы говорим про неопределенность, то, конечно, прежде всего это развитие таких качеств, как гибкость и креативность, поскольку если случается что-то, к чему мы не готовы, от чего мы испытываем стресс, напряжение из-за ригидности, из-за того, что у нас четко прописаны планы, да, жесткое расписание, и не просто оно у нас жесткое, мы не можем вырваться, а еще и психически мы настроены на то, что мы запрограммировали так. Да, и в этом смысле развитие гибкости, вот такой вот способности быстро менять свои планы, чтобы не произошло, да, вот это вот очень важный такой подход. Да, себя в этом смысле вот мониторить. Ну и, конечно, развитие проактивности и креативности, постоянно задавая себе вопрос, а что еще я могу сделать в этой ситуации? Да, я могу упасть и говорить, все провалилось, да, животные не приехали, да, вот конец нашим экспериментам. Да, нет, я задаю себе вопрос, что мы можем сделать, кто мне может помочь, какой может быть креативный ход и так далее. Да, вот мы развиваем, это один из путей, да, сейчас в двух словах, собственно, про то, как говорят, каким образом вот этот вот выкошевый мир может нам помогать и быть выгодным. Мы как раз можем развить очень сильно свои гибкости и свои креативные навыки, да, нахождения, решения проблем в самых сложных ситуациях, потому что каждый день нам нужно что-то такое в этом смысле искать. Да, наконец, по поводу сна, если мы говорим именно про обучение и про работу с информацией, то если мы перегружены, то это нам мешает хорошо заснуть, да, нормально. Если мы не обеспечиваем нормальную область глубокого сна, информация не переваривается, то есть она не усваивается, фактически тогда мы проделываем много работы бесполезно, да, потеря такая времени и плюс еще опять-таки истощение. Поэтому, конечно же, коррекция сна и обеспечение себя нормальным режимом для того, чтобы мы управляли действительно работой нашего мозга. Большие потоки информации. Конечно же, когда мы работаем постоянно интеллектуально в высоком режиме, обязательно периоды переключений в течение дня. И периоды коротких переключений, в которых вы не занимаетесь работой, вы не занимаетесь никаким напряжением, не ставите себе цели, а это могут быть очень простые, но очень действенные практики. Это может быть короткая медитация на 1 минуту, фокусировка на собственном дыхании, да, но она в любом случае, да, собирает и подготавливает вас к дальнейшей деятельности. Это могут быть разные практики дыхания. Если в течение дня, то они могут быть очень короткие, так называемый треугольник дыхания, квадрат дыхания, знаете, да, наверное, но это ровное, например. Треугольник дыхания – это на счет 6 вдохнуть, на счет 6 задержать дыхание, на счет 6 выдохнуть. И так сделать несколько раз, там 5-6 раз. Квадрат дыхания то же самое, только после выдоха мы еще тоже задерживаем дыхание на 6, и тоже мы делаем так 5-6 раз, таким образом мы концентрируем и внимание, и собираемся, да, и силы, и мобилизуемся. И физические упражнения, да, это очень-очень простые практики, вот, но опять-таки эксперименты, опыты показывают, что если это делать постоянно, да, то организм вырабатывает как раз автоматическую конструктивную реакцию мобилизации, да, на вот это, даже если вы уже этого не сделаете и не тратите время на дыхание, да, организм готов к тому, что это происходит. Например, так же, как мы тренируем наши мышцы, это нужно делать регулярно. Если вы регулярно в течение нескольких месяцев занимаетесь нервно-мышечной релаксацией полноценной от 15 до 25 минут, да, то фактически организм перенастраивается для того, чтобы постоянно выносить там большие физические нагрузки. Вот почему рекомендуется очень эти все практики использовать постоянно, да, и вот так вот наращивать эту антистрессовую мышцу. Так, если мы говорим про работу с информацией, да, и для того, чтобы управлять нашим вниманием и быть более продуктивным, конечно, здесь надо задействовать все у нас репрезентативные системы, и визуальную, и кинестетическую, и аудиальную, и еще такую межмодальную, которая уже такая к ментальным практикам относится. И визуально, конечно, это не только чтение, а это составление моделей, рисунки и так далее, мы задействуем это. Аудиальное обязательно друг с другом общаться, слушать, говорить, да, записывать себя там на диктофоны, если есть это, да, практики с тем, чтобы мы актуализировали аудиальный канал. Кинестетический – это когда вы что-то учите, читаете, изучаете и так далее. Это двигаться, это рассказывать истории, примеры, ходить, писать, рисовать, да, то есть вот таким вот образом мобилизовывать фактически все ресурсы мозга и организм, да, запускать. Так, если мы говорим еще про разные виды внимания, то здесь вот скорее написано много, здесь такая установка. Смотрите, человек, который невнимателен, по сути, он не вниманием не обладает, он не обладает контролем над собственным вниманием. Да, произвольное внимание – это контролировать и направлять внимание туда, куда мне нужно. И падение внимания чаще всего бывает вот от того, что мы сказали, по сути, это истощенность. Да. Второй момент, из-за чего мы теряем контроль над нашим вниманием, это наша тревожность. Да, вот очень часто в ситуации стресса действительно наше внимание где-нибудь, а не на том фокусе, который нам нужно делать. Да. И здесь мы берем внимание под контроль с тем, чтобы направлять на задачу, а не вот куда на наши страхи, да, собственно, внимание рассеивается на страхи и тревоги, осознаваемые и неосознаваемые. Вот. Ну и, наконец, мотивация. Да, чем выше наша мотивация что-то сделать, тем больше все-таки мы будем держать под контролем. Если почему-то мы считаем, что ответственность не очень высокая, что это не наша ошибка, что это не наша забота и не наша проблема, да, мотивация падает, а на самом деле ошибка может быть велика, в том числе и для нас. Вот. Поэтому в этом смысле, да, произвольность внимания, необходимо его удерживать и по-разному тоже над этим работать. И прежде всего, вот тревога, которая снижает внимание, это, собственно, разные формы, внешний и внутренний стресс. Да. Стресс – это наша защитная реакция, с одной стороны, да. Это реакция на опасность, мобилизация организма на опасность. С другой стороны, это страшно энергозатратное мероприятие. Вот. И в этом смысле, если мы этим управляем, то фактически эту энергию мы направляем туда, куда надо, а не в разрушение самих себя, что нередко и часто бывает. Да. Если мы говорим о комплексном подходе, по сути, стресс, неважно, чем он вызван, объективной опасностью, нашими собственными там фантазиями, да, это источник опасности. Поэтому стресс – это наша реакция организма. Какую мы должны отправить информацию в мозг с тем, чтобы снять стресс? Что источник опасности под контролем, да, выключить сигнал опасности. Каким образом мы в мозг можем послать информацию, что мы выключили, что сигнала опасности больше нет? Прежде всего, это другая часть мозга – осознание и вербализация. Да? Понять, что со мной происходит, почему я так тревожусь, в чем причина, что я могу делать. Да? И назвать. Считается, если мы переводим эмоционально в когнитивную сферу, да, если мы вербализуем, то у нас уже падение, что у нас мозг заработал, значит, сигнала опасности меньше. Второй момент – физические действия. Да? Мозг в опасности, и мы начинаем производить, ну, там, условно говоря, тигр на нас нападает, мы начинаем его бить. Мы посылаем в мозг сигнал, что у нас есть защита от этой опасности, понимаете, да, физические действия, вот, по сути, такая вот обратная связь. Если мы включаем органы чувств, это вот зарядка, я не знаю, посмотри, найди 5 синих предметов, или посчитай кресло, или обрати внимание на солнце, то есть мы включаем понюхай, чем пахнет, пощупай, чем тепло, холод, какая шершавая поверхность и так далее. Если мы включаем все органы чувств, то мозг получает сигнал, что опасности нет, потому что все нормально. Мы видим цвета, мы видим там поверхности, мы ощущаем и так далее. Это еще один сигнал, да. И многие упражнения по фокусировке внимания, они как раз про это, по очереди пройтись по всем органам чувств, таким образом снизить уровень стресса. И, наконец, общение, близость. Что это означает? Объятие. Мозг получает информацию о том, что у нас есть защита, у нас есть сообщество, которое нас защищает от этого тигра, который на нас напрыгнул. Да? Если мы вот это вот используем, то, по сути, тоже мозг получает информацию, что нормально, опасности нет, можно отпустить, занимайся своим делом. Понимаете, да? Вот этот вот комплексный круг. Если мы подходим так. И в этом смысле... Вот смотрите, у нас стресс в глазах смотрящего. Да, важная новость про то, что мы не всегда, мы далеко не всегда можем контролировать то, что происходит снаружи, к сожалению, да, это не в наших силах, но то, что происходит у нас внутри, мы контролировать можем почти всегда, да, если к этому осознанно относимся. Вот как красота в глазах смотрящего, так и стресс в глазах смотрящего, этим мы и управляем. И у нас есть замечательная фраза Виктора Франкла, да, знаете, наверное, этого великого психолога, философа и психотерапевта, который говорил, у нас есть единственная свобода, по сути, это выбирать реакцию на происходящее. Мы ее выбираем и таким образом двигаемся дальше. Да? И если мы посмотрим на 3 кита стрессового отклика или алгоритм, да, у нас получается первый шаг. Мы ловим стресс, очень главное его ловить сразу, пока он не вырос такой, что действительно им уже мы не можем управлять, нам уже, я не знаю, нужны успокаивающие таблетки или что-то, да, какие-то более сильные вещи. Мы его ловим на каком-то там страх не вот такой, а где-то вот такой. Дальше мы переводим сразу в когнитивную воссознавающую область. Да, что это такое, что со мной, почему, откуда, в чем причины. Вот, когда мы включаем логический инструмент, эмоциональный у нас снижается, по сути. Это может быть страх, это может быть бешеный гнев, да, он может быть вот здесь, а может быть уже такой, что я уже не могу управлять ситуацией, да, и разрушаю вот все, что можно, да, я отлавливаю и анализирую. Переход в анализ, он действительно снижает вот эту эмоциональную вибрацию. Ну и после этого я как бы понимаю, что, зачем и почему, и выбираю какую-то из подходящих реакций. И вот здесь, да, у нас целый спектр и физические, и объятия там и так далее, да, переключения самые разные, возможность с этим работать. И вот еще про осознание, да, я выбираю отклик, вот он управляемый стрессовый отклик, что я чувствую, что я думаю, что я делаю. Да? И на меня наваливается стресс, я чувствую панику, да? Я чувствую в этой ситуации панику. А что я выбираю чувствовать? Я выбираю чувствовать себя, оставаться спокойным, да, и решать свои задачи. Можно, да, себе говорить просто, что я думаю в этой ситуации. Я думаю, ой, боже мой, вообще все пропало, конец света, дальше бежать некуда. Нет, я выбираю думать то, что можно купить билет, можно переслать этих крыс или как там, мышей, да, можно найти другого человека, он поедет, да, я начинаю думать на то, что, какую задачу выполнять. Что я делаю в этой ситуации? Автоматически я сижу, обхватив голову руками, да, и думаю, ну все так. А что я выбираю делать? Я обзваниваю людей, я ищу, да, варианты действий, покупки билетов, отправки, да, решения этой ситуации. Вот, если вот таким образом я себя переключаю на уровне чувств, мыслей и действий, да, то таким образом, по сути, я выхожу из стресса и действительно перехожу к решению ситуации, но в рамках тех, которые, в принципе, возможны. Понимаете, да? Так. И от управления стрессом, вот такое короткое за несколько минут, и еще важный момент, о чем мы говорили, формирование вокруг себя позитивной среды, экологичное окружение, да, с тем, чтобы, несмотря на то, что происходило, все равно вот эта вот среда была позитивная. И здесь можно опереться на модель психологического благополучия Селигмана Мартина, да, это основатель позитивной психологии, который составил те элементы, с которыми тоже можно мониторить каждый этот элемент и все время спрашивать, что у меня есть, а чего нет, что добавить, да, чего не хватает, с тем, чтобы вот эта среда благополучно создавалась. Вот. И здесь обязательно позитивные эмоции, хорошие впечатления. Это могут быть просто маленькие повседневные удовольствия, которые нам приносят эти позитивные эмоции. Второе, обязательно вовлеченность в то, что ты делаешь каждый день. Да, и если, например, любимой работой почему-то учебой не дают заниматься или обстоятельства, понять, каким образом, да, все равно вернуться к этому. Хорошие отношения, про это все понятно, мы этим, да, по жизни занимаемся, но всегда непросто. Обязательно обретать цель, смысл, насыщенность существования, то есть понимать, что, зачем, почему, а не просто, что я занимаюсь там любимой работой, да, немножко подниматься больше, как бы для чего, ради чего все это существует. Ну и, наконец, достижения. И вот с достижениями тоже не все просто, поскольку часто мы страдаем тем, что только большие достижения для нас являются чем-то. Да? И если мы достигли, там, получили большой кубок, большую звезду, там медаль, да, мы собой довольны. Пока мы к этому шли, мы уже растеряли все. Вот. И в этом смысле мозг работает так, мне вам рассказывает, да, что если он получает маленькие награды, то он прекрасный, но все равно большая, огромная звезда или маленькая награда с тем, чтобы он мог спокойно двигаться дальше. Поэтому если у вас огромная работа длительная, да, в которой надо и вообще концов не видно, постоянные коротенькие шаги и все время себя благодарить за то, что я вот это вот сделал. То есть вот примерно как, я не знаю, в социально-политических, экономических, простите мне эти параллели, изменениях. Если хоть что-то чуть-чуть изменилось там в традиционной системе, то это уже большой результат, уже хлопаем, уже аплодисменты, уже здорово, что немножко мы продвинулись. Вот точно так же в работе с собой. Да? Маленькие обязательно благодарим, понимаем, что это достижение, мне медаль, я молодец. Да? Вот как сейчас это делается. Вот, с тем, чтобы мозг получал и спокойно двигался дальше. И очень важно, вот смотрите, здесь вот стоп, сразу стоп, это лозунги убийцы по-русски называется. А вместо сердца пламенный мотор. Это те установки, которые нам транслируют социум, и вроде бы они очень хорошие на самом деле установки. Да? Но если они являются вот такими вот максималистскими, то они являются изнутри разрушающими установками. Да? То есть, например, будьте позитивными всегда, чтобы не случилось, а негативные эмоции никто не отменял, понимаете, да? То есть невозможно носить этот face smile, да, вместо, когда у тебя, например, реальные какие-то объективные вещи. То есть без максимализма. Работайте с максимальной отдачей, каждую задачу, как будто это главная задача в жизни, да? Мы не роботы, да, у нас на самом деле есть много своего человеческого, и поэтому это хорошая установка, да, но надо понимать, да, что вот без вот такого фанатизма, будьте в любой жизненной ситуации сильным, непобедимым, да, вот таким абсолютно идеальным вот таким вот роботом, да, тоже надо понимать, что здесь я могу быть идеальным, а здесь я могу быть не идеальным. Стремитесь к большему, постоянно повышайте планку достижений. Вот это вот болезнь достигаторов, которые все время не удовлетворены тем, что есть, и на самом деле, сколько бы достижений не было, все равно есть полная пустота, фрустрация, неудовлетворенность и так далее, которые мы себя разрушаем, хотя у нас гора достижений, понимаете, да? Выполняйте все со скоростью света, но это совсем, да, это то, что плевать, что есть у каждого свои ритмы, свои нормальные, да, подходы, надо всем бежать как сумасшедшим, забывая просто действительно свою гармонию, которую никто не отменял. Надо жить в своем ритме и свой ритм считать тем нормальным ритмом, который вот подходит, понимаете, да, тоже себя слушать и так далее. Вот еще одна вещь – это тотальная ответственность вообще за все на свете. Да, это я несу ответственность, что где-то взрывы, где-то что-то, где-то и так далее. Кроме того, что я несу ответственность за свою жизнь, за свою семью, за свою работу, за свои результаты. Да, и здесь тоже очень хорошо понимать, что действительно мое, за что я отвечаю, за что я тревожусь, да, а что я воспринимаю как некий греческий хор, те обстоятельства, которые есть, да, вот как бы, и они есть. Я не считаю, что я виноват во всех грехах, которые происходят. Вот. Ну и, наконец, перфекционизм, недопускание ошибок. Да? Ошибка высока, да, ну вот не хочется пускать в параллели всего, в чем мы живем последние несколько лет, да, но вот это тоже вот показывает, да, какие-то даже катастрофические ошибки, да, все равно как бы нужно понимать, что не надо стремиться к этому перфекционизму, это тоже еще один некий разрушительный регулятор, а не конструктивный регулятор. Да, надо стремиться не совершать ошибок, да, но не корить себя, если что-то такое ужасное происходит. И если мы говорим про жизненный баланс, да, колесо баланса, то мы обязательно выстраиваем баланс работы, учебы, жизни, спорта. То есть тоже мы думаем о том, что мы устроены эмоционально, интеллектуально, социально, политически. И вот по каждому из нашему сегменту у нас должна быть наполненность. Да? Если у нас перекосы какие-то, где-то перекос в работу, где-то у нас перекос в чувства, например, а где-то там в интеллектуальную действие, нет. И вот если мы балансируем, ну, естественно, со своими ценностями, да, где-то, что мы любим больше, мы уделяем больше внимания, но на самом деле вот человек, он является вот таким многосоставным элементом, и каждому из них нужно уделять большое внимание. То, что надо уделять физике и здоровью, да, последний вот тренд здорового образа жизни, тут уже никого не оставляет тоже в покое, да, обязательно. Вот. И в этом смысле насыщать свою жизнь, выстраивать колесо баланса как нужно, а также насыщать просто большим количеством маленьких удовольствий, практики, постоянных маленьких радостей, да, с тем, чтобы восстанавливать ту биохимию, которая вот как бы благоприятствует всему, да, тому, что происходит. Так. И, наконец, вот хорошая фраза про позитивное мышление. Некоторые мысли нужно отстреливать еще на подлете к голове. Да, это к вопросу о том, что, конечно, мы относимся ко всему происходящему позитивно и конструктивно. Что это означает? Что означает, во-первых, мы во всем можем найти позитивную сторону и возможности. Там плохая погода, что в этом хорошего, какие в этом возможности? Доллар взлетел, окей, что в этом хорошего, какие в этом возможности? Да, я не знаю, случился бешеный конфликт, что в этом хорошего, какие возможности? Я испортила вином, там облила себе новые костюмы, новое платье, что в этом хорошего, какие есть в этом возможности? Если мы себе все время задаем эти вопросы, у нас получается вот как бы тренажер, да, и мы выращиваем у себя мышцу позитивного, конструктивного мышления. Вопрос, какие в этом возможности, означает, что мы не просто в позитиве, а то, что мы в активной позиции, то, что мы можем изменить, понимаете, да, что я могу изменить, чтобы ситуация стала лучше, если ее надо менять. Вот что означает позитивное мышление. Это не просто вот такой ложный позитив, что все хорошо, да, бомбы на нас летят, а мы все очень рады. Нет, конечно, это совсем не это, видите, вырывается, невозможно обойти эту сторону, да. Что я могу сделать в эту сторону? Вот позитивное мышление. Да, и так же, как и все, позитивное мышление – это привычка, вырабатываемая привычка. Если мы вот так вот мыслим, то мозг у нас дальше без нашего усилия, да, вот если то мы делаем усилия. Вот так все случилось, надо вот совершить усилия и помыслить позитивно, да, если мы это совершаем, то мозг автоматически начинает смотреть, что есть позитивного, автоматически начинает видеть возможности, понимаете, да? И дальше уже нам не нужно совершать усилия, а мы настроили мозг таким образом, если мы постоянно это делаем. Ну вот в целом, наверное, это все, что я хотела вам рассказать. И теперь мы все это вспоминаем еще раз. Да, вот еще раз. Себя, мониторинг – это элементы по вот этому коктейлю стрессоустойчивости и жизнестойкости, да, прям оценка от 1 до 10, что у меня есть, над чем мне надо работать. Точно так же оценка от 1 до 10 по модели жизненного баланса и благополучия, да, а также вот эти вот все тренажеры, по сути, это то, что нас настраивает на то, чтобы мы себя так научили и дальше себя отпустили, уже мы работаем сами на себя, да, а не наоборот вот на кого-то другого, разрушая, давая свою энергию. Вот в целом это все, что я хотела сказать. И литература, на которую я опиралась, это Селигман, это психология, позитивная психология, это Насип Талиб, антихрупкость. Это авторы «Уход за мозгом», очень практическая книжка, которая прям вот рецепты практических упражнений на каждый день. Ну и Канеман, да, двойное мышление, мышление быстрое и мышление медленное, и каким образом принимать решения в самых сложных ситуациях. И Судзуки тоже девочка, которая полюбила мозг, тоже очень-очень практичный прям сборник упражнений. Ну вот, собственно, все, что я хотела вам рассказать. Вопросы? Аплодисменты и вопросы.
Спасибо большое, Екатерина. Вы прям про меня рассказали. Вот абсолютно в какой-то момент у меня работа, как бы выполнение задач, которые как бы сам перед собой поставил, стал занимать там почти все время. Даже стал заходить на сон. Не говоря уже о том, что все остальные активности были уменьшены практически. Спасибо большое, мне было очень полезно. Я уже к этому подошел самостоятельно, что нужно менять образ жизни и менять именно вот в таком ключе. уже у меня начинает появляться там больше спорта, чем это было в последние годы. И, собственно, все то, что вы прописываете, ну, это все не подходит. Спасибо большое, очень полезно.
Супер, вдохновение. Все непросто, но все меняется. Да, мы вкладываем усилия.
Ну вот смотрите, у меня такой вопрос, действительно, все хорошо, очень позитивно, но вот как вот действительно каждый день себя контролировать и запутываться, не отойти. То есть мы как бы всегда есть тенденция как вот к старым привычкам, да, как вот надо себя...
Ну, абсолютно, поэтому смотрите, вот вспоминаю вот эти лозунги убийцы, ни в коем случае не надо себе ставить задачу, что вот я с понедельника вообще являюсь идеальным человеком. Нет. Если я там, не знаю, допустим, негативное восприятие или что-то, с понедельника я начинаю улыбаться чаще, чем я улыбаюсь. Понимаете, мы ставим себе маленькие задачи, и мы их планируем точно так же, как наши задачи рабочие. Там мне надо прочитать столько-то литературы к такому-то числу. Вот к такому-то числу я выработаю, там, я не знаю, 5 минут в день улыбаться. Мы ставим маленькие задачки, мы их выполняем. Когда мы чувствуем изменения есть, мы ставим себе, прибавляем другие задачи. Никаких сверхнасилия никакого над собой не надо. Ну и, короче, как это не забыть, то, что вы говорите, конечно, так же, как и с рабочими задачами, все визуализируется, записывается, наглядно вешается, ничего не надо держать в голове. Мозг должен быть свободен для оперативных задач, конечно. Красиво. Колесо баланса, да, если вы его красиво себе нарисуете, может, кто-то делал, да, и оно перед вами висит, вдохновляет. Достижения. Вот есть же очень такой хороший инструмент, дневник достижений. И вообще рекомендуется его делать вообще каждый день. Но я мало видела людей, которые каждый день делают. То есть каждый день 10-15 минут вечером фиксации конкретной, лучше письменно, да, что важного, полезного для себя, для мира, там, для человечества я сделал сегодня.
Ну, хотя бы раз в месяц, я не знаю.
Ну, раз в месяц, да, но смотрите, вот почему мониторинг очень хорош, если мы делаем это раз каждый день. Тогда мы через месяц, ну, кроме того, что у нас выработается привычка просто позитивно оценивать свои достижения, через месяц просто мы видим на самом деле карту того, что мы делали, что мы не делали. Мы можем посчитать, можно рационально относиться, посчитать, какой процент этих достижений связан действительно с вашими целями, задачами и вашими желаниями, а какой процент связан с тем, что вообще вам не нужно было делать. Понимаете, это своего тоже рода анализ приоритетов, тайм-менеджмента и так далее. Помимо того позитива, который, когда в конце месяца вы смотрите карту, боже, огромный список, неужели все это я? Это же классно. Так что без фанатизма по чуть-чуть, да, ставить задачи. Вот как раз поэтому рациональный подход, если мы линейкой измеряем каждый элемент, мы выбираем один из элементов и думаем, что в течение месяца я подниму этот элемент там, на 2-3 позиции, понимаете, да, вот так вот. Дальше получилось, идем дальше. Так, еще вопросы?
Спасибо, я просто комментарий про мультизадачность, то есть мне прям очень понравилось, потому что ну, вот прям чувствуется падение работоспособности, когда ну, то есть очень много задач разного, там, одна операционная, другая научная, то есть они когда со всех сторон подлетают, но с другой стороны есть тех людей, которые ну, хорошо действуют под давлением. Вот я вот этот феномен хотела спросить, потому что очень много можно ну, копить эту кинетическую энергию, да, а потом за 15 минут, ну, то есть, ладно, конкретно я свой тип описываю, очень быстро, да, делаю. Ну, то есть, и то, что, не скажу, что это плохо, но существует такой тип, насколько он вреден, или это, в принципе, может быть даже физиологично для некоторых людей.
Смотрите, ну в нашей культуре, у нас с вами похожая культура, действительно все больны дедлайнами, то ли это школа, то ли вуз, и действительно, в общем, в дедлайнах поднимается энергия. И это вопрос самонаблюдения. Если на протяжении действительно длительных периодов высочайших нагрузок, то есть имеется в виду 2, 3, 4 года, когда вы под прессом постоянных нагрузок, и действительно у вас организм выносливый вот к этому вот привык, если это выработанная привычка, то окей. Если это вот такая вот амплитуда, то нет, мы не заметим, как рано или поздно это приведет к истощению, к психосоматическим заболеваниям и так далее. Поэтому тут в этом смысле вот нужно смотреть. Вот против амплитуд как бы протестует наш организм, которому вот нужно все-таки балансировать. Да? Поэтому да, но правда в том, что действительно, если мозгу
психологи настаивают на этом и проверяют, поставить задачу, а потом ее забыть, то мозг ее не забывает. Он действительно над ней работает, потом вы актуализируете то, что казалось у вас в голове нет. В последний момент действительно вытаскиваете из головы. Да, но если вы активно этому все-таки регулярно уделяете внимание, то то, что вы в конце вытащите из головы, будет, конечно, несравнимо, да, с тем, что вот просто там в последний момент вытащится. Поэтому в этом смысле капать каждый день, да, вот эта практика, каждый день немножко добавлять. Ну вот стопроцентный всем известный пример с английским языком. Каждый день по одному слову, да, и так далее. Вот каждый день чуть-чуть, и тогда количество переходит в качество с информацией. Так.
Еще один вопрос родился из, собственно, комментария Юлии, вашего ответа. А как быть руководителем подразделения? Получается, вы говорите, амплитуду это плохо, да? То есть начальник должен постоянно держать напряжение своих сотрудников, чтобы они там без амплитуды работали, то есть там не подпихивать их, не пушить все время, а как бы держать на уровне.
Слушайте, ну тогда, извините, это вопрос не про управление стрессом, это вопрос про планирование, организация, мотивация, контроль и управление по KPI, да, ключевые показатели эффективности. Каждый знает свои ключевые показатели эффективности, каждый знает точки контроля, контролирует себя сам, и если потом в конце начальник устраивает разнос, каждый знает, что вот он как бы не доделал то, что ему по... Вот точно так же, да, как я говорю, показатели эффективности мы себе ставим, и к ним идем. Вот точно так же, собственно, это управление. Да, если у вас система управления через KPI, то она реально показывает мега просто результаты. А то, что важно при этом заботиться о благополучии, о физическом, об эмоциональном, устраивать тимбилдинги и комфорт, и фрукты вместо шоколадных конфет, вот это вот все, то, чему нас учат Google и так далее для сотрудников. Да, вот это все тоже well-being устраивает, да.
Спасибо большое, Екатерина. Уважаемые коллеги, наша сессия завершается, и сегодня завершается наш рабочий день. Завтра продолжаем. Спасибо.
Спасибо.
Да.